is out, and I have to apologize, Jonas, because to be honest, and I don't have a good excuse, but I just realized yesterday night that I have two presentations today. I was like, I know I said yes, but I didn't know when. What are you drinking? What is
Malbec.
it?
Malbec. Do you remember Malbec?
Yeah, I have 7 minutes to open a bottle. So it's so nice to see you, Stanley.
Same here, Ricardo. It's great to see you and Jonas and everybody else in Brazil.
I only see Jonas. I don't see anybody, so I don't know who am I avoiding to say hi.
Dr. Ricardo, Dr. Vallejo.
Can you call me Ricardo? I don't like doctors. I try to avoid these guys.
Ricardo.
I know that Ricardo has a meaning in Brazil, or Ricardinho.
Ricardinho.
Okay.
Ricardão.
Oh, puxa.
So for this lecture, you will talk about science of neuromodulation, a journey from Pré-Clinic science to trials.
You're not talking to me. I'm talking DRG. Is Ricardo?
No, to Ricardo.
Oh, Ricardo. Okay, all right.
Let me see, because I know I prepared a presentation, but I cannot find it now.
We can talk about neuroscience and technology.
Well, no, but you know, the thing, Jonas, gosh, what I did with my presentations. Wow. What I'm trying to do, gosh, this is, so I have one of them is here. I put it in a poster called Brazil. So obviously it should be there. No, what I was thinking, Jonas, I have two presentations.
Okay.
So this first one, I'm gonna be talking more about my journey, my own journey. That's kind of the way I envision that. From how did I started studying preclinical studies, and how did I ended up translating it into the clinical trial, showing some of the results of the RCT that we just finished. That I think they are already, I already presented it for Brazil. And that's it. That's what I plan to go there. And then the next presentation for the next meeting, I'm going to go more into mechanism of action, trying to avoid overlap between the two of them, which is kind of hard because large part of my journey has been in preclinical and clinical. So is that okay, Jonas?
Yes. The first lecture is correct.
Okay.
You will talk about your journey, your clinical journey.
Perfect.
Perfect.
Hold on, I just realized I don't have this presentation. So, Stanley, I'm very curious, my friend.
Yes, sir.
So you are drinking Malbec and coffee?
Oh, this is water.
Oh, you have to be healthy. You have to dilute the resveratrol. Okay, I got it. Now, what is my time, Jonas? Because I just put some slides together, and I want to be respectful with your time. Or
8:55 Brazilian time.
No, but no. What is my time? How much time do I have?
How many minutes? 20 minutes.
20. Okay.
20.
I'm just gonna speak faster.
Jonas, is there a timer at the bottom of the screen?
You can make your time. We are here for you.
No, I'm saying, but is there a timer at the bottom?
No, there's no timer.
There's no timer. Okay. All right.
There's some overlapping rooms.
Right.
The rooms are called like Ricardo Vallejo, Stanley Golovac, Jonas Lenzi, and the lecture starts at the right time.
Okay, but
You
there,
can go.
but after
How
me,
many
are you
years
speaking, Jonas, after me?
What?
Are you talking after me?
No.
No,
I'm here to
to moderate. Okay.
yes, to moderate Ricardo's lecture.
Who is the moderator?
I am.
Okay. So if you don't mind, please take 10 minutes away from Stanley and give them to me. Just
He thinks that I don't bring value to the table. Well,
you bring so much value, Stanley. I just think that you are so good that you can present much faster than I can.
Porque eu posso falar português e o português fala rápido, né?
Yeah, well, you can speak Portuguese if you want, but I just wanted you to do it quickly.
Yes, sir.
Now, I am having trouble here to see you guys.
Oh, boy.
Stanley.
Hey, boa noite, como é que vai? Tudo legal?
Boa noite, Stanley. Pô, que imenso prazer estar com você aqui.
O prazer é meu, honestamente. Uma grande coisa para eu estar aqui com vocês. E eu queria estar no Brasil, é lógico, mas com o COVID nós não podemos viajar frequentemente ainda não, mas já pronto.
É uma honra.
Kill me, Stanley.
Sir.
Can you stop pretending that you speak Portuguese, please?
I'll speak slower for you Spaniards to be able to understand.
Stanley, it's a great honor for us, and you know we would like that we are all together, but we are together in our hearts, and so I hope everybody in your family is good, and we are going to have a good section here.
Oh, yeah, absolutely. Very interactive.
And
I don't see my camera. Okay. Oh, here I am. Okay. I exist. How many more minutes before I can?
Vallejo, how are you, my friend?
Leandro, I'm really sorry. I know I have been a nightmare for you. The only thing I can say is Stanley knows me well. I'm not a nightmare for you. I'm a nightmare for everybody. So I'm not doing it for you. I'm really sorry, Leandro.
No problem,
so
my friend. You are on time, and we are talking with Stanley in a few minutes. So it's a great honor for us that you are between us, and you know. For us, you are the heroes of interventional pain medicine, and so we're going to have a so good section, and I would like to talk with Stanley that if he wants to start the presentation, if you want, okay. We have the first one. Just a minute. Stanley is the first one.
I have a question, guys. I don't know how to share my screen. I'm sorry. I think that Stanley already knows that. I don't.
Don't worry, my friend. And you are in the when you see a plus under the name Vambu.
Under what? I'm sorry?
A plus sign just below Vambu logo.
Okay.
Can you find it?
Yes.
Yes.
So you can see share when you have a share the presentation. Yeah.
I have presentation. I have stop sharing, but I'm not sharing anything as far as I know.
Oh yeah, because Stanley is going to start, and in a few minutes you go. Yeah.
So I follow Stanley?
No, you follow Dr. Manuel Jacobson.
So is Stanley?
Manuel and you.
Manuel and me. Okay, so I can put a jacket and look professional. Okay.
So good, my friend. So Stanley, you go ahead.
Can you see my screen?
Yes, perfectly.
Wow, boa noite a todos. Espero que todo mundo está bem no Brasil e no mundo inteiro. Aqui nos Estados Unidos estamos em COVID lockdown, but estamos bem. E muito obrigado a José Campos, e a Jonas e todo mundo no board e o panel que me está permitindo falar com vocês hoje de noite. Vai ser um pouco de portunhol, né? Me desculpa, mas não tem maneira de falar DRG em algumas maneiras em português. So, going back on the history of where DRG started and how this all evolved, we've been using electricity to help stimulate the spinal cord for many years, and all of us know this. And throughout the years, we've been able to pioneer different things to be done. Some of them have worked very well, and some of them have not worked very well. For me, fortunately, I started back in 1985 doing spinal cord stimulation. So I started way back in the early days with one company being Medtronic, 4 contacts. We initially did this. We thought this was sliced bread. We thought this was the best thing, que nós podíamos usar eletrodo com uma bateria e era fantástico. Mas, de repente, tinha 3 companhias mais, né? Boston Scientific, A&S came into play, and Medtronic, and we started learning more and more that, maybe if we increase the contacts, if we increase the vertebral body zone and area, we could possibly capture more of an area of pain. Then we started talking about battery technology. We had challenges with primary cell, and we needed more energy and more energy and more energy, but we couldn't get it because the battery would last 3 years, 4 years. So we started talking about what else could we do, and that would be a rechargeable battery, more leads, better programming, and hopefully we were able to show that. Then all of a sudden in 2013 to 15, another company came out. Maybe we don't need paresthesia. Maybe we can have a paresthesia-free type of a stimulator. And we thought, well, okay, that's going to use a lot of energy, and it does use a lot of energy. So now we're in 2016 to 2020 and beyond. Where are we? Well, we're talking about DRG. Why are we talking about the dorsal root ganglion? I'll get into that in just a moment. This is now becoming true modulation. We're not just altering it electrically. We're talking about chemical alteration, which Giancarlo Barilat, the great neurosurgeon in Denver, has always taught me. It's not spinal cord stimulation. It's spinal cord modulation. But if we can modulate other structures, maybe we have something there that should be able to improve our patient's outcome. And especially talking about low energy, because energy is the exhaustion of using a primary cell battery. So why do we talk about the DRG? Well, back in 2003, Ken Allo, in 1993, Ken Allo and I were in a lab together. And Ken said to me, you know what, maybe we should try doing something a little different and not just putting the electrode over the spinal cord. What do you think about going out the foramen? And I looked at Ken and I thought he was absolutely crazy. I said, my goodness, you're going to take an electrode that's 1.3 millimeters in diameter and you're going to force it out the DRG entry zone? That was incredible. Well, we did it. And the reason it captured such a great amount of improvement was the area has the least amount of cerebral spinal fluid, which is a huge conductor of electricity, as you know. So this shows here where the DRG is located. As you can see, it's in the superior part of the foramen. And so your electrode, when we show how it's placed, should come over the top of the actual DRG. The concentration of cells, of which my good friend and colleague, Dr. Vallejo, is going to talk soon, is about glial cell. And the glial cell seems to be the whole crux of where all this composition of cellular activity occurs. If we can introduce electricity prior to getting to the spinal cord, we may have the answer there. So CRPS seems to be a devastating entity and a devastating phenomena. None of us really know exactly how it actually develops and who develops it more so than others, but it does occur. There's a psychopathological area that has to be involved. There's areas of nerve injury, such as causalgia or trauma to the nerve itself. There is central sensitization, where the individual becomes depressed. There are areas where there are altered sympathetic outflow. And these inflammatory changes all stand to cause a significant problem. So if we can look at where in the entire schematic this would be involved at conducting electricity to stop the energy from getting to the dorsal column, we look at the dorsal root ganglion. And that area is extremely important because in the middle of the dorsal root ganglion is the area that we call the T junction. And that T junction is like a traffic light. And if you can stop it from propagating the electrical conduction to the cord, we may have the answer as to solving individuals' pain a little bit more easier than we typically do. When we talk about DRG, let's talk about stimulation is designed to address the limits of what conventional spinal cord stimulation does. Number one, we all know that it's somewhat of an unstable area. It's a large zone. It's an area where there is lead migration, and that can be challenging. So we have to be able to anchor properly and hope that the area does not cause our lead to move, because this could be devastating for everybody. The next area is unspecific stimulation. How many of our patients have told us that we need to only cover a specific dermatome, but yet the spinal cord gives them the entire leg and the portion of their buttocks and a portion of their hip, more than what they bargained for? So this can be problematic for certain patients. When we go into the energy usage, we know that 75% of the entire energy in a lead that's placed in the spinal canal is wasted. 75% when you use a cylindrical lead. Only the unidirectional paddle lead is the one that can show that the energy goes in a downward displacement. So high voltage and high energy use is wasted when we do spinal cord. The Accurate study was something that I had the privilege of being a part of, myself and 20 other centers throughout the United States. And what it did was it tried to determine specifically, making a comparison with spinal cord stimulation, whether DRG would be beneficial or not. So we had 152 patients that were enrolled in the study. We had a control arm that had spinal cord stimulation, and we had other subjects that were randomized in order to only have DRG. We had 22 investigational sites, 3-month primary endpoint, and we used causalgia and CRPS as the causative factor of where the diagnosis was in order to help these individuals. Lower limb was defined as anatomy below the iliac crest, except the axial low back pain of the area on the superior crest itself, and the subregions identified as the areas of the knee, the ankle, and the foot of whether they had pain. So this showed, as a typical common endpoint, greater than 50% pain relief, as we all expect, greater pain relief, and also at 3 months post-implant, with the freedom to stimulate the area induced by neurological deficits if they chose not to like the type of stimulation they were receiving. What we found out was an incredible amount of improvement. We saw that the patient reported the area of pain was in the low back, transcending down the leg, or in the ankle and the foot. Some individuals were in the knee itself. Some individuals were more of the entire leg. The therapy areas also demonstrated, with this great work from Dr. Chakravarti, that the area not only is electrically inducing the change inside the canal, but it also is altering the pro-inflammatory changes also. So this is telling us about modulation, not just electrical stimulation. And this is why it's so important, because this is now translating into modulating the chemical factors like tissue necrosing factor that is devastating to causing vasoconstriction inside the canal. So here we have an anatomical dissection. We're showing the schematic here of the spinal ganglion, which is the dorsal root. As the propagation of the impulse goes into the dorsal column, it goes on the lateral track and then all the way up to the hypothalamus. So what we want to try to do is to inhibit the somatosensory or the sympathetic reflex, because remember, these individuals have a significant amount of edema, pseudomotor changes that cause the area to be dysfunctional, changes that cause the area to be non-useful. So if we can mitigate edema, we can improve function, we can create an area that is more rewarding to the patient to look more normal, the patient returns back to be in a normal state of functionality, if you will. When we talk about the disease states of CRPS, this is a devastating injury. It's almost 5.5 to 26 per 100,000 individuals will succumb to this unfortunate disease. CRPS is about 3.8 to 8.3% after a fracture. Individuals are placed in a cast, and they have limited range of motion, which causes atrophic changes to the musculature and atrophy to the muscles and the joints themselves. This creates joint stiffness and joint pain, which then leads into a plethora of other changes that we will discuss momentarily. When we look at a study that was done by Kemmler, it's a very interesting one. It showed that traditional spinal cord stimulation achieved at best 50% improvement. You know, I hate to use these numbers of 50% because we think that that's a milestone, but you have to also remember that almost half of your individuals and half of your patients fail to improve. So I think we should set our standards higher than 50%. The potential reason for Kemmler's outcome was with peripheral nerve damage. Remember, the damage is not in the cord, the damage in the peripheral area that causes the initial impulse to go to the DRG and then to the cord and to the hypothalamus. So these are the areas that concern us the most. The highest concentration of cellular activity along with the glial cells is found in the dorsal root ganglion. So why not stimulate the area? If you're able to put an electrode directly over the area, you can quiet the noise, and quieting the noise is what we're trying to do with all of our stimulation. Lessen the impulse that is interpreted by the brain. So why is it a predictable location? Well, much like a spinal cord stimulator, you have to place this inside the canal. You have to think about maybe I'm there, maybe I'm not. Some of us do something called trolling, where we start at T7 and T8 and T9, and we lower the electrode until the patient tells us, you know what, this is the sweet spot. This seems to be the area. But this doesn't happen with DRG. We know that T12 is T12. We know L3 is L3. When you place that electrode right over the target, you know specifically that that area is covering that specific dermatome. When we look at the area now that I'm showing you on the screen, the organization is very important to new work that I'm going to discuss momentarily by Ken Chapman. Ken has gone off and started to do some work at looking at specifically just low back. And low back, as we know, with traditional spinal cord stimulation, is very difficult to achieve. It's not as easy as one would think. But if you go to the T11, T12, and L1 complex and put 2 electrodes out each one of the foramen onto the DRG, you may be able to encompass the low back phenomena that have eluded us for so many years. So these are areas to be concerned with, because when we've done our dissection and we've done our mapping, we go back and look, and the lower back area seems to be in the more lateral area of cuneatus and gracilis. It's not in the center where we tend to place our electrode. So thinking about that, going back to the slide just one more time, if you go to T12 and you place in the electrode at the DRG, that is why we tend to encompass more of the lower back area. So it's a phenomena to think about. When we initially designed the system, it was called an axiom system. It had a programmer, it had a larger battery, it had a sheath, it had the introducing needles, which are 14 gauge 2E needles, and then it had a stylet and a specific electrode. We've now moved on to something that is now called the Proclaim or rechargeable system, which is a little bit more user-friendly. To perform this procedure, it's the standard prone of the patient. We try to mitigate the lordosis and flatten out the back. We try to allow the entry of the needle to be somewhat like 30 degrees, and typically accessing the epidural space. But unlike a traditional paramedian approach, this one has to be from the opposite side. So with the target being on the right side, you want to come in from the left. You want to come in from the left, 1 to 2 segments below the targeted area. And then you want to be able to thread the actual sheath all the way up to the foramen. So this is showing us the anatomy of L4 and L5. Typical entry zone is 1 to 2 levels below. The L5 one is slightly more challenging, obviously, because of the iliac crest. So things that are not as that used to be easy to perform with this type of procedure is slightly more challenging. When we go up to the T10 and T11 area, once again, we come 1 to 2 levels below, and we try to target the area of entry in order to go out the neuroforamen. So the device has several pieces to it. There is a sheath, there is a connector for you to be able to inject some saline. There is a stylet inside the actual electrode, which helps to stiffen and to help to push the electrode out the neuroforamen. So when you get close to the neuroforamen, you tend to tickle or tend to stimulate the area, and you have to push through the supporting ligaments. This allows for the electrode to be pretty fixated inside the epidural space. In addition to that, what we're going to do is create what's called a strain relief loop, and I'll show you that in just a moment. So the sheath delivery system is a protector. It protects the electrode, and within the electrode is a stylet that is sitting within. So once the needle is placed in the epidural space and it's angled towards the neuroforamen, we then deploy the sheath that holds the actual electrode and the stylet. Once it reaches a foramen, you continue to advance the electrode forward as it goes through the ligament and out the neuroforamen. We typically like to do 2 to 3 contacts on top of the DRG in order to ensure that enough contacts are on top of the targeted area that we want to stimulate. Once inside the epidural space, we'll then pull back on the sheath. The sheath is shown here as being pulled back by this little indicator, and this is what's called the strain relief area that we're going to perform. So we're going to create a loop inside the epidural space, and once that's created, it looks like an S inside. And typically speaking, we've had very, very little migration, if any at all. There is also another anchor that is going to attach itself to the electrode, but for the majority of the time, the lead does not move. Some of us have even done some cervical type of transforaminal DRG stimulation for individuals that have had surgery and have had ongoing persistent pain. But these areas are a little bit more challenging, remembering that the spinal cord is in front of the needle there, with the cervical enlargement being at C5, C6, and C7. A unilateral approach is sometimes used also for the area that is difficult at L5 and S1 because of the challenge of the iliac crest. So this allows us to push the lead outside of the neuroforamen from a unilateral approach. So final reminders for us to be able to contemplate whether we should be using DRG or not. Should we use general anesthesia or MAC anesthesia or sedation or local? We tend to use only local. We like to know if our patients are experiencing pain throughout the procedure. We inject 4 to 5 cc's of normal saline in order to help expand the space. The introducer is fairly rigid, so it does cause some difficulty and some challenges with some discomfort. So every once in a while, the patient will complain of some discomfort. Migration is usually not expected, but we do anchor our leads, and we tend to fixate it with a ligament approach of trying to secure the anchor to the ligament. The DRG, as we all know, is an important structure generating and maintaining chronic pain. DRG T-junction is the area that we really concentrate where the stimulation occurs. Modulation of different neural fibers is frequently dependent in that area, such as the glial cells, and DRG stimulation has been shown to modulate these neuroinflammatory markers, which for all of us now is game-changing and not just an electrical field that is used to stimulate the area, but also neuroinflammatory markers. Quero dizer muito, muito, muito obrigado a todos. Eu vou deixar meus colegas falar agora e mais tarde nós vamos ter perguntas, tá? Boa noite.
Dr. Stanley,
Sim.
eu vou pedir uma gentileza para o senhor.
Sim,
enquanto
como?
a gente abre a outra sala, o Dr. Tiago Freitas, de Brasília, nosso querido amigo, ele perguntou ali, I would like to ask about his experience using spinal electrodes in GRG, comparing to specific electrodes designed to GRGs. Você poderia responder enquanto a gente abre a próxima sala?
Sim. Sou Tiago, boa noite a você e eu espero que você e a família estão bem, e seus pais. This is a challenge. É um pouco mais complicado, né? O diâmetro do eletrodo é 1,3 milímetros. É mais grande que 0,7. Então, quase a duplication of the diameter size. This becomes an issue because of the narrowing of the foramen. If the disc has any kind of disc herniation or disc protrusion that is pushing into the neuroforamen, and you're also placing a larger than what has been deemed to be acceptable as an electrode, you may compromise the neuroforamen, and that may create a stenotic area leading to neurogenic claudication that is iatrogenic. It's not to say that it can't be done, but you have to assure yourself that the diameter of the foramen is large enough to support the structure and potentially any injury to the disc. But that would be my recommendation, that the intradiscal height is large, is non-herniated, and this would give you the best opportunity to stimulate that dermatome.
Thank you very much.
Obrigado, rapaz. Gusto em vê-lo, hein?
Prazer é meu.
É,
Bom demais te ver.
prazer é meu, Tiago. É meu.
Muito obrigado.
Stanley,
Hein?
parabéns pela apresentação. Meu amigo, como sempre, você realmente foi brilhante. Que aula, viu? A gente vai agora para a aula do professor Manuel. A seguir teremos o Dr. Ricardo Vallejo. E se você quiser acompanhar lá, clica no ícone lá, Manuel Jacobs, entra na sala. Depois da sala do Manuel, vocês podem voltar ou ficar naquela sala que, a seguir do Manuel, vai o professor Ricardo Vallejo.
Tá bem. Eu volto, tá?
Tá, e aí a gente tem a discussão ao final da apresentação do Vallejo, tá bom?
Perfeito, né? Um abraço grande.
Parabéns novamente, muito obrigado pelo aceito do convite, viu? Foi uma honra pra gente.
O honor é meu. Muito obrigado, hein? Tchau,
Valeu.
tchau.
Até breve.
Até breve.
Essa sala do Vallejo
the bamboo. Yeah, I'm here, but I was listening to Stanley's presentation. He finished, you talk, and then everything went blank. I don't see anything. I see you as presenter. Hold on. It seemed like I'm alone. I see you. It says presenters LB, but I don't see anybody else. I saw Tiago was there for a minute ago. I'm sorry, Leandro.
No problem.
Okay.
Como é que faz para you? Just a minute. You can go out and entry again.
I cannot hear you well through the screen. Now I can see Stanley again.
Oh, I'm there. Sorry.
You can see Ricardo Vallejo. Beside the Carlos Moro and Philip Peng.
For whatever my camera. Okay, now my camera is.
Well, I'm looking for you.
Okay. Yeah. So what do I do next, Leandro? I'm getting anxious, and I don't like anxiety. What do I mean, my screen doesn't show me how am I going to be presenting my slides. I assume somebody's talking right now. My screen again disappeared. I don't know why. As soon as Jose Luis, can you hear me, Leandro?
I hear you, Ricardo. Are you on Google or are you on Safari?
I'm on Safari.
No, they want you on Google Chrome.
Okay, let me log out.
Yeah. They want you on Google Chrome.
Ok.
Hi.
Tudo bem? Leonardo, tudo bem? Tudo fantástico.
Está?
Ei, como é que vai? Tudo bem?
Tudo bom. Estou tentando colocar o Vallejo aqui.
Aí, Stanley.
Como vai, Stanley?
Cadê o Vallejo? Saiu?
Ei, rapaz.
Ele vai entrar no Google Chrome agora.
Ele vai tratar de entrar no Google Chrome.
Yeah. Stanley, aí, tudo bem, meu amigo? Família, você melhorou do coração?
Rapaz. Eu tô cortado aqui, mas eu tô bem. Tô bem. Montei 40 milhas hoje na bicicleta. Tô bem.
Beleza, 40 milhas, hein?
Parabéns,
rapaz,
parabéns.
Tem que dar duro, rapaz. O que é isso?
Olha, vê o Vallejo entrou aí, o Zé.
Deixa eu ver se ele entra aqui.
Ele apareceu ele aí, ó.
Apareceu, ele vai entrar. Deixa eu habilitar ele. Ele já vai entrar.
There you go.
There you go.
Hey, hi, my friend.
Hi, my friend.
Why you like Apple?
You are so cute now, huh?
Now, yes, of course. Now you can see me.
Ricardo, who came to the rescue here? I just want to know who came to the rescue.
You did. Yeah. I owed you a back.
Yes.
I know how to buy you.
Oh, you do? You do.
Yeah. How are you, Ricardo?
You know how to buy me.
Okay.
So what is going on right now? Is somebody presenting?
Now we are going to let me see. Manuel is presenting, but let me see your time.
He's in a different room.
In 2 minutes. 2 minutes if you want. In 2 minutes, if you want to start, you can start. Or okay.
I don't understand this system, but okay. I'm gonna start in 2 minutes. And where am I? Why has Manuel finished already?
Not yet, but you in 2050 you can start your presentation, and Manuel is going to continue the presentation in another room. All right?
Okay. Am I gonna see you after my presentation? I'm getting a little bit nervous now.
So you see the bamboo name on the left side.
Share screen.
Yeah. Yes. Now you are sharing. I think so.
Am I?
Diz pro pessoal, quem quiser vai começar lá no Vallejo e quem quiser continuar com o Manuel, continua com o Manuel, e se não, vá para a sala do Vallejo, tá?
Você escreve o seu texto.
Vai lá, escreve lá. Entra na sala do Manuel.
Can you see my screen?
Let me see. Not yet.
Well, I am.
Try another time.
Now I only see myself, and I don't have the bamboo. I don't have the bamboo plus anymore.
Yeah,
I saw it. Sorry, I saw it. I was on top of that share screen. When I said share screen, your entire screen.
Como é que tira esse negócio daqui? Não, ele quer compartilhar a tela dele.
Quem?
O Vallejo.
Valeu? Manda ele clicar no mais.
Ó.
Can
No mais.
I don't see the screen. Can you see it?
Ele fala português?
Fala.
Manda no canto superior.
Ó.
No canto superior, acima.
I think. Let's see.
Não, ele tá faltando.
Tá carregando a tela.
Não, tá faltando ele colocar o aplicativo por baixo. Clicar no aplicativo.
Que aplicativo?
No PowerPoint.
Vallejo.
Yes.
You have to in your screen your presentation in the background of your computer.
It is in the background of my computer.
Put it on the desktop, Ricardo. Ricardo, on the desktop.
It is on the desktop, but when I say share a screen,
and then just click on the presentation.
Yes, it's sharing, but doesn't charge. Yeah.
Is it there now?
No, it says loading.
Not yet. It's loading. Yeah.
Okay. Why do you guys seem to know this system and I don't?
No problem. It's
so
different
it says
for everyone.
Share your screen, and it says your entire screen, application window, or Chrome tab. I put it, share my screen with my whole entire screen. And again, in my background, I have my presentation in presentation mode. I put share, and it calls me and says sharing blocked by your browser. Okay.
Well, you need to go to system preferences. You go to your system preferences.
Yes.
You need to unlock and allow Google to.
This is so complicated. Hold on.
No.
System preferences, and I have to go where, Stanley?
I'm looking.
And
sharing.
safety, privacy.
Sharing on the privacy.
Privacy. Yes.
Privacy. Where's privacy?
Security and privacy. Okay.
Yeah, security and privacy. Right. Put your password in.
It's not asking me for my password. This is an Apple, my friend. It's a
No, I have an Apple. You can't unlock the little lock on the bottom left. You have to unlock it. It's going to require a password.
Yes.
It's an Apple, my friend.
Okay, whatever.
Thank you, Stan.
Oh,
Now what do I have to do?
So try to
So
share.
now go to full disk access on the left-hand side. Click it
You mean in the
dialog box.
Okay.
It says full disk access. Click it, and then you should click Google Chrome on the right.
Hold on, where? In my system preference?
Yeah.
Yes.
System preferences on the privacy, Ricardo.
Uh-huh.
Now go down to the little lock.
Não precisa ele botar que é
The
preferência.
little lock
It is
on the
open
left-hand
already.
side. Click it.
I already put my password,
Okay.
Stanley.
Now on the left side of your screen, you should have location services, contacts, calendars, reminders. You see that?
I have contacts, calendar,
Yeah, scroll
reminders,
down.
photos. I don't see Chrome.
No, scroll down. No, scroll down to full disk access. Scroll.
Okay.
Touch it.
I did it.
Okay, now on the right side, you're going to have many things. Scroll down until Google Chrome, and then Check.
It says Google software update.
No, do Google Chrome. Keep scrolling. You should have Google Chrome.
No, there is no Google Chrome. I
You
have
don't have Google
Cisco.
Chrome?
I have Microsoft Outlook. I
Right.
have Cisco, Zoom.
Stanley,
Sim.
eu acho que é na parte de gravação de tela.
Eu acho também.
Record screen.
On the record screen.
Em record screen, Ricardo,
Yeah.
você vai ver...
Screen recording. Go
Yes.
back down to
Record
on the
screen.
left-hand side. Underneath your full disk access, you should have screen recording.
Yes.
Click it.
Full
no,
screen recording.
I
Okay.
send one picture for you, Ricardo.
Screen
Okay,
recording.
I have Google Chrome will not be able to record the content of your screen until it is quit. So that means I have to quit Google Chrome.
You should be able to click it and say no, and it should be okay. But if
It
not,
says
you may have to
later or quit and reopen.
You have to
Later.
quit and reopen. El español está complicado, ¿eh?
Sim, muito.
Saiu, Zé. Ele saiu. Ele reiniciou.
Não
Tá,
tem
mas é normal.
jeito, rapaz. O cara não sabe dar jeito.
Não, mas agora deve estar aprendendo, Stanley. Está
É.
aprendendo.
Ai, que
Ok.
coisa. Esses
Eu preciso ir numa outra sala, pessoal. Só uma licença.
Oh, my friend Ricardo. Ay, ay, ay. What am I going to do with him?
It's not so simple to present Dr. Ricardo.
No, it's not. It's not.
Wow.
Ay.
I will take 5 minutes to introduce him.
Okay.
Ricardo, you're there. Okay.
Now, do I have to try to share my screen again?
Yes, I would.
Yes.
I would think you should try to go to screen sharing on the system preferences, and then you should go to Google Chrome. It should be checked.
Hold on. I don't like when you tell me go back to system preferences. I just wanted to start my sharing of the screen.
I can't help you there, Ricardo. I can't help you.
You could have come here or something and help me. Okay. I
It's
am
too
here.
cold. What is it, 40 degrees out there?
Screen.
There you go.
Can you see something?
Well, it says
Loading.
loading again. You like to load a lot. There you go, Ricardo.
Now, are you seeing yourself in my screen, or is you are seeing only my screen? Because I'm seeing I see in the middle of the screen there is the screen, which I can make bigger or smaller.
If
Can
you
you see my
start your presentation, it will show up on the screen.
Okay.
Okay?
So we have 20 more minutes, I heard.
Well, you only have 2 minutes now because you wasted your time.
You have your entire time.
Okay. So what do we do now? Sorry.
I need to introduce you to our audience.
Okay.
We take 5 minutes. Eu convido todos agora para ouvir a palestra do Dr. Ricardo Vallejo, uma pessoa muito ocupada, muito estudiosa, que não tem tempo de ficar gastando com esses computadores aí. Ele está mais na fase pré-clínica, ele vai explicar sobre a sua jornada. Ele é diretor de pesquisa do Millennium Pain Center, está profundamente empenhado em descobrir novos métodos e tratamentos para dor crônica. Ele é membro do conselho editorial de periódicos como Clinical Journal of Pain, Pain Physician, a Revista Mexicana de Anestesiologia, atual editor associado da Pain Practice, membro da banca examinadora da UIP, professora adjunto da Illinois State University. E o Dr. Ricardo tem uma experiência muito grande e eu não quero apresentar totalmente ele, porque senão vai faltar tempo para a sua brilhante apresentação. The screen is all yours, and we are so happy to have you here with us.
Thank you, Jonas. As you said, I'm not very good with this computer stuff, but thank you very much for helping me. And it's a real pleasure to be with you all. I was asked to talk about the journey from preclinical to clinical trials. So when I think about a journey, I think about my own journey. So that's what I'm going to be trying to tell you, what I have done over the years. And that comes back with one of my favorite movies is the Groundhog Day. And I don't know if you remember this movie, but it keeps repeating and repeating the same day over and over again. And I think it is pertinent because it kind of tells you where my journey started. It was back in 2004, the first year that, by the way, that I went to Brazil. I was invited by Luciano Brown to go to Gramado, Brazil. And I was extremely excited, as you can imagine, to go to Brazil. But that was the same year that I was started my real interest into the spinal cord stimulation area. And there were two aspects that I think are important to describe in the way I felt back in those days. In one hand, I was very excited because I saw amazing response from patients that had failed all other forms of therapy. But at the same time, I was extremely frustrated because my patients, after a while, did not respond correctly. And I was trying to understand what was going on. Was it poor patient selection, or was it something else going on in this particular situation where I had to face as a clinician my patients and they were not doing as good as I was hoping? And if you can see here in the left side of the screen, this is a drawing from Patrick Wall to Ronald Melsack, where he describes how the conduction of nerves can be modulated. In this case, how the modulation of nociceptive signals can happen if you activate large myelinated fibers. Since then, there has been a total focus into how can we optimize these parameters to obtain the best possible modulation of the nociceptive signals. I would say that over the last decade, there has been amazing advances in terms of the technology that allow us to use a wide range of frequencies. We can use from 20 hertz all the way up to 10 kilohertz. We can use bursts with passive recharge balancing, with active recharge balancing, and we can use different amplitudes. We can go from subthreshold to a paresthesia-based stimulation. That is totally different than what used to happen back in the 1990s or back in the early 2000s when I started my pain practice. Because back in those days, life was simple. You just had to only understand the gate control theory. You had to understand that you had to modulate the A beta fibers in such a way that your contacts should be placed in a location to generate those paresthesias, and then that should prevent the patient from being in pain. But unfortunately, these things were not working, as I just mentioned. Around 2015, a big change in this paradigm came. I was part of the Ciencia trial where we evaluated the use of a paresthesia paradigm, paresthesia-free paradigm, trying to alleviate patients' pain. And interestingly, those patients responded in the Ciencia trial, but that brings a major consideration. And is that if you look before that time, all the different approaches were based on computational models. So if you think about the spinal cord, all these different parameters were optimized to try to generate, based on computational models, based on mathematical formulation, the best possible activation of the A beta fibers. So when you are considering that, you can come up with all these different arrays of contacts, always trying to target these neurons. And as you can see here in this drawing, in this cartoon of the neurons, you can see these neurons connecting with thousands of different synapses, transmitted the information from one to each other. I like to to make reference to this particular poem by Wallace Stevens. He was a wealth management in Connecticut, and he, in his free time, he used to write poems. And in one of his poems, he writes about this mystery that happens in the language, which in order to decipher the meaning of a particular sentence, you have to decipher what is the meaning of each one of the words. But even when you achieve that, when you understand the meaning of each one of these words, they don't give you the right context of the meaning of the whole phrase. And I think it is very important, as I was trying back in 2004 to reflect on my frustration with my patients and what can I do different to try to provide better relief. And my thought back in those days was that I had to go back to a basic understanding. What am I stimulating? Am I stimulating neurons only, or there is something else that is going on, something else that can lead to this particular pathological pain that our patients are experiencing? And then I took a radical approach in which I first tried to understand what was going on in terms of the pain. You cannot treat a condition unless you understand what condition you are treating. In my case, I was trying to treat chronic pain. That was the disease. I was not trying to generate a different programming algorithm that will fulfill the gate control theory or that will somehow just focus in the modulation of the neurons. I thought that it was fundamental to understand what was I treating, a disease-centric approach in which I could understand the disease first. And the first glimpse that there was something beyond just the gate control theory and that there was a much more complex system leading to the pathological pain that my patients were experiencing was probably formulated in 1994 by Garrison. And you can see in this animal model how in this initial, in the A box, you can see how these animals had a section of the spine. They had no injury at all. And this is the way it looks. But if you had a chronic constriction injury, a damage of the sciatic nerve, ipsilateral to the injury, you could of astrocytic cells, which are marked by a marker called GFAP. So if you apply a NMDA receptor antagonist, MK801, you will see how not only there is a reversal in terms of the behavior of the animal towards pain, but there was also a dramatic decrease in the activity of these glial cells. So that back in those days created the concept that in order to have pain, you needed more than just the neurons. So from this original drawing from Linda Watkins, where she just refers this painful stimulation going through the nerves into the spinal cord, we were also able to observe that there was something else missing. Back in the 1990s, for those of you that remember, there was a big emphasis in research on patients with septicemia. And you can see how viruses or bacterial infections were able to generate an activation of the microglial cells that will lead to the release of massive amounts of pro-inflammatory cytokines that were going to generate in the rodent model a behavior that resembles pain. Now, back in those days, though, everybody just thought about the glial cells as supporting structures with no function in any neurodegenerative condition or even in the pathology that I was looking, which was pain. There was this missing aspect in terms of what was going on with pain. We have to understand first that when you have a small injury, there is going to be some glutamate release at the level of the synapse, in which glutamate is going to interact with AMPA kainate receptors and other ionotropic receptors to transmit this chemical information and transform it into an action potential. And then we have learned since 1965, when the gate control theory was formulated, we have learned that the astrocytes play a pivotal role not only in the development of pain, but also in the maintenance of pain. What happens when the astrocytes are kind of in a resting state is that the glutamate is taken out of the synaptic cleft. They are metabolized within the astrocytes, converted into glutamine, and recycled into the neurons for the neurons to produce GABA or glutamate. Now, it's important when we think about these cells that these cells have different functions. For example, the microglial cell is sensing the environment, and it has neurotransmitter release receptors that can observe whether or not there is a massive release of neurotransmitters or any foreign object or substance. But the astrocytes also play a pivotal role in maintaining the homeostasis. Something that is very unique is that one single astrocyte can provide coverage, can envelop the synaptic clefts up to a 140,000 synapses. So keep that in mind. There is one cell that can somehow synchronize or coordinate the neuronal conduction, and it has this amazing ability to coordinate hundreds of thousands of different synapses. Now, as the pain continues, there is going to be massive release of calcium gene-related peptides, substance P, glutamate, and at that point, just not only the ionotropic receptors, but also the NMDA receptor become activated, and you can see how all of them, after generate phosphorylation of intracellular kinases, which lead to further activation of the receptors. And something that I want to focus today on is a massive increase in calcium, which comes from the endoplasmic reticulum. And also there is an activation of the microglial cells and the astrocytes in such a way that the astrocyte no longer maintains the homeostasis, not always remove the glutamate out of the synapse, and this persistent glutamate concentration in the synapse is going to end up in the production of reactive oxygen species and nitric oxide. At this point, these glial cells will also start releasing pro-inflammatory cytokines, and they will be critical in the development and maintenance of pain at the level of the neuroinflammation, amplification, modulation, and in case of pain, the distortion of these afferent signals. After considering the condition that we are treating at a molecular level, my next question was, what are we treating? When we are placing the electrodes in the posterior aspect of the spinal cord, what are we really treating? So we did a study in Spain with University of Valencia, and what we observed is that there is a dramatic increase in terms of the cells population. There is a marked overpopulation from glial cells in relationship to neurons, in such a way that immediately underneath your electrodes in the surface of the dorsal columns and the dorsal horn, the ratio of glial cells to neurons represents 12:1. That means that 12 cells are underneath your electrical signal for each neuron. That represents about 92 to 95% of all the cellular population underneath your electrical field is a glial cell, which means that it doesn't matter how do you want to approach the problem, these cells will be exposed to the electrical signal. It doesn't really matter which type of electrical signal you are applying. It will always be some type of effect on these cells. Now, let's consider what I gathered back in 2004 when I was trying to come into studying the effects of electrical signals on different biological processes. First of all, and it's quite obvious, the glial cells have a different resting membrane potential than neurons. The second aspect is glial cells do not develop action potentials. But when they get exposed to electrical signals, they respond by modulation of these biological processes that I just mentioned to you, and they respond differently to different frequencies, different amplitudes. And as you can see in this center image, they also respond with glutamate release to different ways of recharging your balance, whether you do it actively or you do it passively. And also, when they respond to electrical signals, they respond by calcium waves. And calcium waves are extremely important. I mentioned it in the previous slide, because they can modulate very fast conduction around astrocytes. There is these hemi channels or gap junctions that can connect a massive amount of astrocytes that can communicate like if they were one single cell by these calcium waves. When you think about it, that means that those islands of astrocytes can control or coordinate the communication of hundreds and hundreds of thousands of different neurons. This is one of the first publications that we did when we applied conventional stimulation to rats after nerve injury. And what we observed at a molecular level is that genes involved with inflammatory response or immune-related were upregulated by these currents, and this work has been recently corroborated by the group in Johns Hopkins, where they did a similar study with a different animal model, also using conventional parameters, and what they observed was, again, the same modulation of genes that were involved into immune and inflammatory response, as well as downregulation of the synaptic signaling cascades. You may be wondering, okay, you can modulate electrically the genes, but at the end of the day, the main effectors in biology are proteins. So we did this study where we correlated our response in terms of gene expression with protein expression, and as you can see, we obtained a high correlation coefficient between these two values. This is just one example in vivo, where we simply, this is a correlogram, and the colors represent the different expression, the correlation between different genes as they are exposed to no electrical signals, actively balanced signals that are symmetric or asymmetric. And you can see just by changing one of the variables in the electrical signal, you will be able to modulate the co-expression of genes that are involved in immune function, in glial modulation, and in redox reactions. So that takes us into an important aspect. The way that I wanted to approach the problem was, what do electrical signals do to the neural tissues that are exposed, the glial and the neurons? And in order to do that, what I did is I look at what is an electrical signal. Everybody can say, oh, I have a new waveform. In reality, if you think about it, a waveform is just basically 4 components. You have a frequency, you have a pulse width, you have an amplitude, and you have a way to recharge your balance, whether it's active, passive, symmetric, or asymmetric. Those are all the variables that you have. You can repeat them 10,000 times, or you can repeat them 50 times, but it's the same type of electrical signal. Now, when you think about it, the way I wanted to approach the problem was, what happens when I look at each one of these variables independently? So I just show you here what happens when I keep the frequency, the pulse width, the amplitude, all of them constant, while I modulate only the recharge balancing. The same way I approach different frequencies. If I wanted to evaluate frequencies, I only modulate frequencies maintaining the same pulse width, the same amplitude, and the same recharge balancing. Based on that, I observed that there were some different biological processes, some of the ones that I just mentioned, that responded differently to each one of these 4 variables. By doing that, what I decided was that we had to combine these different signals, trying to target the different cell populations in a multiplex signal and observe what was the effect of that. What happened at the animal levels? When we compare it with high frequency at 1200 hertz or low frequency at 50 hertz, the combination of signals, and keep in mind there is here more than 32 different combinations of signals that we tested before we came up with this particular combination, provide a much better improvement in behavior for mechanical and also for thermal hypersensitivity. And the same response was observed when we jumped from rodents into sheep animals. When I look into neuroinflammatory mediators, this is a heat map representing genes that are involved in neuroinflammation, those that magnify and lead to the pathological processes at the microglial cells and the neurons. And that what we observe is that by combining these different signals, we were able to revert 78% of neuroinflammatory signals involved in the neurons with a high correlation coefficient, as well as 79% of microglial genes. Now, I had mentioned earlier the calcium, and the reason I want to mention that is because I want to take you a little bit of a deeper level. This is the first time that I'm presented internationally, this data, but I am talking now about proteins. So we know that we can modulate genes involved into the neuroinflammatory process, but what happens at the molecular level with the effectors, the proteins? And we all know that ion regulation across cell membranes plays a critical role in cell communication at the neuroglial interaction and will activate intracellular cascades that are involved in activation of metabotropic, ionotropic receptors, and at the end of the day, the modulation of transcript factors. And I mentioned calcium because increasing intracellular calcium is one of the main inducers of neuroinflammation, and this propagation of calcium, as I explained earlier, will lead to the communication among astrocytes and the coordination of electrical signals around about thousands of different synapses. And this is what we observed. We observed that by combining these different signals, we were able to reverse IP3, is inositol 3-phosphate, which is the main molecule that leads to the release of calcium from the endoplasmic reticulum. But we were also able to revert all these calcium cascades that were happening in such a way that we were able to reduce the intracellular calcium concentrations. And that happens not only at the level of the neurons, but when we look at specific proteins for astrocytes, we also observe similar effects. Now, one of the first things that I mentioned is that all this process that has been taking us so far 16 years and we are still moving forward came out of my frustration with my clinical results. At the end of the day, I'm a clinician and I wanted to treat my patients. So out of these efforts, we started formulating different combinations of parameters that ended up in this randomized controlled trial, which is a multi-center trial at 12 different sites in the United States, in which we were trying to control axial low back pain. The purpose of the study was to determine in a non-inferiority analysis whether or not the combination of therapies, nowadays called differential target multiplex signaling, was better than conventional stimulation to control axial low back pain. 128 patients were randomized, 94 of those were implanted, and the primary outcome was at 3 months. After analysis of non-inferiority, then a superiority analysis was done for axial low back pain as well as for leg pain. And very soon in January, we're going to be releasing the secondary outcomes for function and disability of these patients. So the randomization was 1-to-1. And when you look at the demographics of the patients, one good news, patients were quite old, an average of 61 years of age. And you can see how these patients also had pain for a long duration. And the reason I'm saying that this is good news is because it mimics some other previous studies. Probably this has been the oldest patient population that has been studied in a randomized controlled trial. But that means that even for elderly patients, there is good hope that they may respond to spinal cord stimulation. So when we compare differential target multiplex with conventional stimulation at 3 months, we obtain non-inferiority, and then superiority analysis was performed. And when we look at intention to treat analysis, in other words, every single patient that was randomized was included in the analysis, the responder rate, those patients with at least 50% relief were 80% in the DTM versus 51% in the control. These results were maintained up to a year. Again, we look at every single patient that went through the trial. That's what we define as modified intention to treat analysis. 84% of the patients in the DTM still had more than 50% relief. This is the mean pain at 1 year, 1.74 below 2. And when we look at profound responders, defined as those with at least 80% relief, we observed that 35% of them in the control arm, but 7 out of 10 of the patients in the DTM group obtained more than 80% of axial low back pain. And I want to emphasize this axial low back pain because that was the main purpose of the study. When we look at leg pain, also, the majority of the patients, the mean pain was 1.45, less than 2 at 12 months follow-up. So in conclusion, we had 84% of the patients reporting more than 50% relief of axial low back pain at 12 months. We had 7 out of 10 of these patients reporting more than 80% relief, and the mean pain at the end of the study was 1.74. There is a lot of talking right now about different mechanism of action. Some people say, oh, I can do this or I can do that. You know, I can put my lead here or I can put my lead there to try to obtain the best results. If somebody tells me, well, how does it work? It reminds me this famous movie called The Time Machine. It was originally, the book was written by Howard Wells, and in a movie in 1960 by Rod Taylor, he is asked at some point, how do you go to the future or how do you go to the past? And his answer was very simple. If you want to go to the future, you just move this label forward. If you want to go to the past, you just pull it backward. We need to understand the mechanism of action if we want to optimize the therapy for our patients. So that has been my journey, and I thank everybody again for this invitation. I wish I had been, like I do almost every year, in Brazil one more time, but for the time being, I wish you the best, and thank you very much.
Thank you so much, Ricardo. It's a wonderful area. I know your service, and it's a wonderful work that you have been done during the last years. So thank you so much for your presentation. It was wonderful. So I would like to start the conversation about this topic, and I'd like to pass the word to Dr. Jonas and Dr. Lear, and for me, and we are going to go some questions about the presentations. And after the discussion, we are going to stay in this room because we are going to continue in the topic of neuromodulation with the Dr. Caporal and the second class of Dr. Vallejo, and the last one, the Dr. Tiago. So if anybody wants to do any questions for the speakers, you can do that right now.
Thank you,
Tiago. Hi, Tiago.
Hello, Dr. Vallejo.
How are you doing, my friend?
Pretty good, thank you very much. Well, I made 2 small questions, but I have a third one just to discussion. So, in this trial about DTM, that 1 year follow up, these patients, most of them were Pfeil-Baxter syndrome, or they were just back pain originally without surgery. I would like to know about that, just to know.
Yeah. So let me answer that, Tiago, because when you asked two small questions, I got very worried. I like this last question. It seemed like very straight. So yes, the patient were patients with back pain. The large majority of the patients were patients with previous back surgery. They represented 80% of the patients. Now, the question is what happened with the other 20% of the patients? What I can tell you about that, Tiago, is that we did an analysis, a separate analysis, and we were so excited about the results that we are currently conducting two randomized controlled trials, one in Europe, one in US, in which we are using it exclusively for patients with axial low back pain and virgin back. I'm sorry, axial low back pain with or without leg pain and virgin back. And the reason we do that, I published a paper in 2005 on patients with discogenic pain that I treated with a spinal cord stimulation, and the results were very promising, but I never had a team that could help me to run a large study. And I think there is a big need for those patients to have the indication of a spinal cord stimulation before they are exposed to surgeries that not always are positive. So that is my work for the next 4 or 5 years will be induced in these two randomized controlled trials. And we'll see if it works. But I don't know if that was your question, Tiago, in terms of what else beyond the failed back syndrome.
Yeah, exactly. Just because of the population that you use it for this paper, and it could be used for patients that did not have surgery. So that's a real challenge sometimes. You do a lot of stuff, a lot of procedures, and patients don't get better.
Yes,
so
nice. I tell you, we are very, very excited with the results that we observed in the RCT. And we are right now close to 30 patients enrolled in the randomized controlled trial. We have, at this point, I think 18 sites open for enrollment. It has been a little bit slower than what we hoped because of the COVID, but we are moving forward. Europe is also, we are doing a multinational study in Spain, as you can imagine, obviously, also in Germany, the Netherlands, and Belgium. We hope to be done with enrollment by middle of next year for both studies. One is against conventional medical management. And the reason, some of you may argue why, the reason was because for reimbursement purposes in Europe, the government entities selected conventional medical management. In Europe, in US, we are comparing it with conventional stimulation.
Okay.
Now, you have two little questions, and I'm trying to understand them, Tiago. One is waveforms in spinal cord stimulation really modulate the glial cells. Absolutely. The answer is yes in rodents. In humans, well, no. It's unethical to take spinal cords from humans. But I very briefly mentioned, hopefully I will have more time in my next presentation, but if you look at the correlation or reproduction of the studies in rats, when we look into the ovine model, they are pretty much the same. You observe exactly the same amount of relief. And there is, the anatomical study that we did in terms of cell population was in humans. And I have hard time to believe that from an evolutionary perspective, cells will differentially respond. The astrocytic cells are way more complex in humans, dramatically more complex. I would say that there is a lot of research nowadays in Alzheimer's, depression, and other neurodegenerative conditions where they have demonstrated that the astrocytes and glial cells in particular probably have a lot more to do with memory, with cognition, with consciousness, pain, obviously, and modulation of other sensory signals into our brains.
Thank you very much. Thank you.
Ricardo, the two, or I only respond to one?
Ricardo, I have two small questions. It's not so simple to understand the underlying mechanisms of chronic pain. Since they have been approached by DTM. Did it happen any neuroplastic phenomenon?
In the randomized control trial, I have no idea. We did publish, though, well, we haven't published. We are presented it as an abstract at NANS, and we already presented it as an abstract at ASRA. It won the best basic science award, in which we showed that we could modulate long-term potentiation and other forms of neuroplasticity. So absolutely, if you are now, the question is whether you are modulating these pathways in the right direction or not. But yes, you definitely interact with neuronal plasticity.
The second one, does DTM alters any suffering supraspinal mechanisms or only spinal cord mechanisms?
Well, that's an interesting question. I am not so lucky as Stanley. Despite I tried, I wasn't able to get my wine to respond to this question appropriately. But I would say the following. Let's say that you have right now back pain, and I try a therapy that I'm 100% sure is gonna alleviate your back pain, at least temporarily. My experiment consists on taking you to the MRI suite. I'm going to do a functional MRI to evaluate the function of your brain. And I will make a comment about functional MRI in a moment, by the way. But beyond that, I will take some type of hammer. And what you are laying in the table, I'm going to hammer your tibia. I guarantee you that you will have no back pain. And I guarantee you that there is going to be activation of central supraspinal centers in your spine. So the question comes into, if the patient gets relief from pain because the stimulation is working and you see changes in the brain, is it because of the effects of the stimulation in the spine, or is it because the patient has less pain? And that is a quite complex question to respond. I am sure that there is effects, what we call orthodromic effects at the central level, the same that there is antidromic effects like vasodilation mediated by calcium gene-related peptide in the periphery. Now, when we talk about this type of supraspinal mechanisms, it's fundamental to understand something. What is called the BOL, that is the blood oxygenated levels that you are observing. What you are observing is an increase in the vasodilation in the areas of the brain. That doesn't mean that there is an increase in neuronal activity leading to an increase in oxygenation or need of oxygen. Because if that was the case, you will see an increase in deoxygenated hemoglobin. And that's not what is happening. What you are observing in the functional MRI is an increase in perfusion. And just as a little mention, perfusion in the central nervous system at the level of the blood-brain barrier is handled by astrocytes, not by neurons. So the neurons have no control whatsoever in what is the amount of blood that is required in a system. That means that there is a connection at the neuroglial interaction between the neuron and the astrocyte. And the neuron tells that something is going on, and the response of the astrocyte is to bring more oxygen. So what is really going on? And again, it's like you mentioned at the beginning, Jonas, everything is very complex. But we as physicians, and I believe that physicians, we are scientists, you can put all the art that you want, but we are scientists at heart. We have to try to unravel this problem. The fact that there is a very beautiful gate control theory that is easy to understand doesn't mean that that's the way nature is. And it is our job to unravel this problem. And I'm sure that I will never do it. But my job is to try to do that. And it doesn't matter how complicated it is. I have to keep peeling and peeling and peeling this problem until one day we get a better understanding. So again, it's not as simple whether the effects are central. What I can tell you though for sure is that when you apply an electrical signal to neural tissues, means neuronal and non-neuronal cells, they are exposed to these signals and they respond locally to that. What happens above or below It's not my area of research, but it's fascinating.
It's so fascinating. The sentence you put it in one slide of your lecture, in the sum of the parts, there are only the parts. And when we understand a small part, this part is continuously changed by the neuroplastic phenomenon.
Yes,
it's so
yes.
difficult.
I like poetry, and I mean, Stanley can testify with a good glass of wine, and I mean, Leandro can do the same thing. We have shared a lot of wine together. I can be a lot more prolific than that.
A lot more poetic.
Doutor Alexandre, tem uma pergunta?
O professor Manuel também tem uma pergunta aqui.
Manuel,
Por favor, professor.
I have a question here from Manuel.
Leandro, você está mudo.
Thank you.
Hi, Manuel. It's great to see you again.
Yes, we have talked about this subject before. We are in an international project to use the spinal cord stimulation to improve the rehabilitation of people with spinal cord injury and sensory and motor deficit. In the past 25 years ago, we were engaged in a project to put a Piplon over the area of the lesion in the spine, in patients with the partial lesion of the spinal cord, to try to improve the rehabilitation. Just one patient from 8 had some accelerated rehabilitation, which was effective, but all of them had improvement of the spasticity with the Piplon. A Piplon presents a very big amount of neurotransmitters. But do you think that with your concept about the effect of the spinal cord stimulation, using your method, we can accelerate the rehabilitation, not just stimulating, but making stimulation in the way by which you are doing your work?
Thank you, Manuel. Excellent question. I will need a freisolada on top of the wine right now to answer that, or pau de queijo at least. But let me, we have a paper that right now is in review, and the main criticism, and I think we're going to get it published very soon, is why we didn't use female rats. And I think right now there is obviously a very clear understanding that the response of stress and pain in females and males are different. And poor rats, I think the female rats are totally against this movement to introduce them into studies, but it's moving in this direction. But what I can tell you is the following. The paper is called The Modulation of Microglial Cells. So to try to make that very simple, what we did is we apply electrical signals with different parameters into the spinal cord. And we observe at microglial cells. And as you may know, and this is an oversimplification, like everything in life, there is a gray light. So it's not like A or B, but we humans try to simplify concepts. So when the microglia is activated by injury, we call the microglial activation into 2 stages. One is called M1, the other one is M2. What we are trying to do in this paper is to mention, like there is a grade scale. There is an M1 going into M2, there is an M2 going into M1. But what is interesting, and you probably know that a lot better than I do, Manuel, is that what causes the real damage after a spinal cord injury, especially partial spinal cord injury, is the activation of the microglial cells with the release of reactive oxygen species, nitric oxide, and all these different proteins that are released in that area. That's what causes the real trauma. Then the M2 type, the good microglia come later to repair the trouble that the M1 already created. What we were able to observe was that electrical signals were able to modulate and kind of discriminate whether you have a predominant M1 or a predominant M2 type of population. So you can have a pro-inflammatory microglia or an anti-inflammatory microglia. We observed very early on that animals that had a spinal cord injury, when you look at microscopically, you will see that there is a higher repair in those animals that were initially stimulated. Now, the practical problem in my mind is the following. Let's say I can demonstrate in an animal model that the early usage, because the only value will be to prevent the M1 to destroy the tissues. So if I can modulate the apply electrical signals to prevent activation of the microglia into the M1 and promote M2, probably most of the damage will be prevented. But I am not a neurosurgeon, but I have hard time to believe that anybody will go after a spinal cord injury and place electrical leads, do a laminectomy and put electrical leads. Maybe some transmagnetic fields or something like this could be used for that particular approach. But I'm guessing. I don't have even wine, so I'm just guessing that it is fascinating. Something that one last point, and I apologize, Jonas, for taking so much time. But something that I was trained originally in Colombia. I did my medical school in Colombia before going back to Spain. And we have, as I suppose you guys have in Brazil, leishmania. And one thing that I was, at some point, I wanted to create, and I'm going to present in the next slide, on the next presentation, something regarding our in vitro experiments. So we were trying to grow glial cells in vitro. It seems like a simple thing to do. Everybody has done it, but as I always have trouble even to connect to Chrome, you can imagine how hard it was for me to grow glial cells in vitro. So I couldn't, and I had already my whole team for a summer already hired and ready to work. So I was able to obtain Leishmania tarantulae, which is the non-infectious form of Leishmania that is used for research. And what I did is, in vitro, I applied electrical signals to this Leishmania. And what we were looking was for motility of the Leishmania and for acid phosphatase, which is an enzyme that prevents the engulfing and destruction of the Leishmania by the macrophage. Interestingly, you could modulate electrically the release of this acid phosphatase. What I'm trying to tell you is something that is so obvious that it probably doesn't need to be mentioned, is that every single cell in our body has a resting membrane potential. It's essential that if you have a membrane, there has to be a resting membrane potential, and therefore they will respond to the application of negative or electrons to the field, and they will respond to the way that you retake those electrons out of the field, whether you leave them there or you immediately create an anodic pulse to regain them out and take them back into the battery. So it's fascinating. There is so many things to look into it. I'm just scratching the surface. Unfortunately, my time is coming to an end, and I hope that researchers, young researchers or fellows like you will continue this area, because just thinking that we only have to focus into neurons, I think has prevented us for 50 years to move this field forward.
Thank you.
And Eli, some questions?
Vallejo.
Hi.
Great lecture. Good to see you again.
Thank you.
I'd like to ask you something. How can I compare failback surgery and a vision spine and both with neuropathic pain?
I'm sorry, can you repeat, Alexander? There was somebody talking. I don't want to say who, but there was somebody, I couldn't hear you well.
How can we compare failback surgery syndrome and a vision spine pain with both with the neuropathic pain?
That's a good question. how can we compare that? So there is a little thing that I didn't mention about the virgin back study that we are doing. In my virgin back study, I am ruling out facet joint pain by medial branch blocks, or at least medial branch block mediated pain, depending how do you want to conceptualize that. We are ruling out sacroiliac joint, and we are ruling out instability. If you think about it, it leaves me with one option. I can, or maybe two. I have muscular pain, myofascial pain, or I can have discogenic, or what is nowadays named vertebrogenic pain. I am very biased since 2005. The reason I did this initial feasibility study for patients with discogenic pain, and I'm going to say something that still sounds bad, is that in my mind, discogenic pain is a sympathetically mediated pain. And I know it sounds horrible because everybody thinks in discogenic pain as a mechanical pain. But I will argue that all the innervation of the disc, especially the lower portion, that is the one that is having the load of weight, is handled by sympathetic nerves. The great Raman communicants are the ones that are providing that, and that's sinovertebral nerves. So that means, in my mind, that sympathetic pain responds quite good to spinal cord stimulation. So I think, in my mind, it will be much better to have a virgin back patient without all these excessive tools in the back, excessive rods and screws in the back, and try to treat that condition. Now, I am the first one that believes that if you have instability, that should be treated by surgery. But beyond that, I think that there is a very gray area. And if it is myself, I will rather try a spinal cord stimulation before anybody puts a screw in my back.
Okay. Ricardo, I have to unfortunately disagree slightly with you. I think that there are neurochemical mediators within the disc that cause neuroinflammatory changes that lead to Alexandra's question about virgin back causing neuroirritation to the sinovertebral nerve, as you indicated. That is not necessarily a mechanical issue. It is more a neuroinflammatory issue that is responded by the sinovertebral irritation. And having done some work inside the disc, and it happens to be my holy grail of perseverance, of trying to determine why is it that neuroinflammatory complex, metalloproteinase, prostaglandins, yada, 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 why are they so problematic? I think has an element that we need to address, not only what you've eloquently stated, but I think that there is neuroinflammatory indicators within the disc that are very caustic, are very irritating, are very problematic to individuals that have back pain that needs to be addressed. But that's my 2 cents.
I totally agree. But again, Stanley, that one of my original predicates is that spinal cord stimulation, when applied correctly, can modulate neuroinflammatory processes. So yes, I totally agree. I agree with you 100%.
But may I comment, please? We know presently that in the osteoarthritic pain, at least 50% is neuropathic.
Yes.
Because there is sprouting, all the mechanisms related to the inflammation, distortion, causes secondary neuropathic pain. And we cannot say that discogenic pain is just inflammatory. It's also neuropathic.
Yes.
As syndrome, as myofascial pain syndrome, and so on. At least 50%.
I agree. I saw your comment. I agree.
Go ahead, Ricardo.
No, I was just saying, Stanley, that I saw Manuel's comment earlier on, and I sent you a message with agreeing and 3, maybe I put 4 exclamation marks. So yes, I totally agree. Jonas, you say Ricardo, but nothing else.
Low back pain is a very complex area to address. It has no single answer. It is very integrated, and it involves the aspects of multiple facets, from discogenic to facetogenic to neurochemical to irritating to, it is a complex phenomenon. So the treatment involves some very high-level decision-making as to how to address. But, Ricardo's work goes unsurmised. To thank him so much for what he's done. And we need to continue to evolve to address this area, because it is the most devastating area in our industrial nations of what we all suffer with.
I agree. I have, guys, okay, thank you, Jonas. I am obsessive compulsive. You just wrote Ricardo and you didn't write anything else. So
we are going to please, Ricardo, if you want to take some water,
yes,
you can.
Fine.
and why sometimes, but if you want to start your second presentation, you may.
Do I have to go to another room? Do I have to
go out? Here right now, here.
I may be here some molecules, not that
because Dr. Leo is coming from other room for join to us, and we are going to your second presentation, and after you have Dr. Tiago, and in the final we can talk a little bit more about neuromodulation. Okay,
so it's Leo first, then Tiago, and then me. So I have a lot of that calls for more than water.
You are first, then Leo. Leo was in another room right now, but he's finishing. But
so
you
can start.
First.
I go first, or he goes first?
He goes first. He's finishing the presentation right now.
Okay, you let me know and I start talking. I'm like a taxi.
Yeah.
Okay.
You may go.
Okay, water. Thank you.
Water.
Foi duro encerrar essa discussão, né?
Mas senão a gente vai até amanhã aqui. Significa, o tema é realmente assim extremamente interessante e assim de um interesse realmente de todos, né? Alguma outra pergunta que alguém possa querer fazer, professor Manuel ou para o Stan?
Uma pergunta que eu acho que vale a pena é a questão do bold, né? Do bold de estimulação. A prática clássica era estimular 1 hora, 4 vezes por dia, uma vez que os equipamentos no passado não tinham capacidade de bateria para alimentar 24 horas. E a gente observava que o resultado era bom, havia um after effect que era de outro, funcionava exatamente como a gente tem agora. Quando a gente faz isso dentro de um ambiente de algum modo nós vamos dar um pouco de folha para fazer. É a questão da intermitência da estimulação e da estimulação contínua. Vou falar agora como neurologista, doença de Parkinson. Quando você desliga o sistema implantado no doente de Parkinson, ou você tira ele dopa 2 vezes por semana, volta a usar, ele sai um foguete, sai bem demais, ele melhora muito a função. Então, folga, um pouco de pós-operação estimulante. Era uma pergunta que eu interessante que ele respondesse. Qual é a opinião dele sobre fazer intermitência da estimulação? É uma que até hoje a empresa a Abbott tem um projeto correndo a respeito de voltar ao passado, fazendo intervalo de repouso. Não sei qual dos colegas. Sandro, precisa ligar o microfone.
Tá ligado já, doutor Manuel. Agora desligou de novo. Liguei aí.
Leandro, ligou. Tá ligado.
Liguei agora, liguei. Professor, você acha que essa intermitência tem a ver com uma fadiga que possa existir de uma estimulação continuada?
Eu tenho a impressão que sim. Eu vejo que a natureza quer repouso, né? Você dorme à noite, quando você dorme, os seus neurotransmissores, a sua vida hormonal se modifica, você nasce de manhã com melhor memória, com maior disposição, com desempenho físico melhor. Eu entendo que isso também deveria ser aplicado à neuromodulação, porque nós não estamos o tempo todo andando, nós estamos o tempo todo ativando um arco reflexo, nós temos um período em que nós descansamos. E a neuromodulação, sob o meu modo de entender, ela devia dar ao doente também essa oportunidade. Mas é o meu modo de pensar, entenda? Não foi provado nada assim.
Bom, nós poderemos manter as nossas discussões posteriormente. Vallejo, se você quiser iniciar a sua apresentação. Opa, eu vou ligar o seu áudio, só um minuto.
So I go. Okay, I go. You ask for it.
You may go.
Okay. Can you guys see my screen? Not yet. Can you see my screen now?
Yes.
Okay. Okay. So, hi everyone. I just finished a minute ago, so this is kind of a continuation of the presentation that I just gave about my journey. And I have to take advantage. I cannot escape this opportunity to say hi to my dear friend, Leo. And I'm going to talk about the mechanism of action from a little bit a different perspective. I will start by one of my initial comments, and is that the gate control theory, as you can see, it only has these lines which represent neurons. And these lines are within a vacuum. There is nothing around that. You already know the result, the rationale that I have, but I think it's worth to mention it because the big question that I think arises is, why do we look for a mechanism of action? What is the value of a mechanism of action? And I think that it is fundamental to have some sort of understanding based on observations of what is happening when you apply electrical signals, because only when you do that, you can generate some working hypotheses that hopefully will lead you to the appropriate response. You can generate some experiments, you can obtain results, and based on that, generate conclusions and further try to understand what is at the end of the problem. If you think about the gate control theory, what it requires is that you apply an electrical signal. This electrical signal we know is more potent at the level of the cathode, and then that will start to attract ions towards this area, creating a depolarization of the neuron, and then at the level of the anode, you will have a hyperpolarization. This is what we learned very early on, again, based on computational models initially designed by patch clamp techniques by Hodgkin and Huxley, who created this conceptual model in an amphibian model. Later on, McIntyre proposed a mammalian model, and nowadays we have amazing computational models that can represent hundreds of neurons. Now, keep in mind that Dr. Hortelano, who is from Brazil, recently discovered that we humans don't have 100 billion neurons, as it was always claimed, but only 86 billion neurons. So those computational models that represent 500 neurons or so, and they are so complex, really do not represent what is going on in nature. Now, if you think again, what is the original hypothesis is that if we apply an electrical signal, and these red dots represent an electrical signal that has enough duration and enough intensity, you will generate an action potential. If it happens that the intensity or the duration of the pulse is not long enough, your resting membrane potential will increase, will become more positive, up to a point that if it doesn't reach the threshold for depolarization, then it's going to go back into the resting membrane potential. Something that is important, though, is that here you are seeing how the neurons are an all or none phenomenon. They will generate an action potential or they will not generate an action potential. But there is not this more female-ish type of response in which you will observe a wide range of responses depending on the different parameters that you apply. This is a very, very old slide. This is from 2006. And in those days, I was formulating that we had to start thinking about electrodynamics and look at the different parameters, current, frequency, pulse width, and recharge balancing in terms of determining what is the effects of these components of the electrical signal into the neurons. Again, nowadays the technology allows us to do a wide range of different things. And I mentioned earlier, in the good old days, life was simple. You knew you had to place an electrode in a particular location that was going to generate depolarization of those neurons, A beta fibers, that will create paresthesias in the painful areas. Because we wanted to stimulate A beta fibers, then it was obvious that you wanted to generate paresthesias. So the amplitude, the location of the electrode was quite simple. And before the rechargeable systems, everybody used passive recharge balance because it was just easier for them and they were going to save energy. Similarly, pulse widths were around 230 because that is what is called the chronaxy. And just a very brief description, what it means is the real base is the amount of current that is necessary for a neuron to depolarize. Now, if you double this amount of current, then you generate a pulse width around 240, and that is what is called the chronaxy. Very commonly used terms back in the early 2000s. But as you can see, all these computational models that I have previously commented on, or these different approaches, were always just aiming to provide depolarization of these neurons. But again, when you look into these neurons, and I think if you close your eyes for a second and you imagine a neuron in your brain, this is what you will imagine, this emptiness, this empty space, and these bright extensions communicating with each other. Now, I will argue that until very recently, what we have done is the following. A patient comes into your practice and then you start trying to determine what is the source of pain. You may try different medications, you may try different injections, and nothing helps. And then suddenly this eureka moment comes into your brain and you decide this is the perfect candidate for spinal cord stimulation. Now, once you have done that, you will arbitrarily choose some parameters and you will hope that these parameters will provide the best possible modulation of the neurons in order to keep your patient happy. That is the approach that we have been following all these years. And what I'm going to try to do today is to formulate a different approach. I will say that the gate control theory created this amazing field of the neuromodulation, but at some point, premature simplifications and systematizations in science could ossify science and prevent vital growth. And I think that that is what unfortunately has happened in our field. We have totally ignored for almost 50 years the amazing developments in the field of neuroscience that give us so much understanding about the most important aspect maybe to consider, which is what are we treating? We should not frame ourselves into these restraints that prevent us to go beyond just modulating the neurons. But that's what the gate control theory has been doing for us all these years. In an earlier presentation, I was mentioning how astrocytes provide extensions that surround the synaptic cleft in such a way that we call it nowadays the three-synapse compartment. In these three synapses, we have the afferent neuron, we have the postsynaptic neuron, and we have the astrocyte, which is again governing the hemostasis at that level. It is the astrocyte, for example, the one that is in charge of retaking the glutamate and generating the glutamine. And believe it or not, when you talk about excitatory or inhibitory neurons, GABA or glutamate, probably you never consider that these cells cannot generate glutamate or GABA without glutamine, and that they are incapable of generating these substances without the fuel of glutamine that is only provided by the astrocytes. Once astrocytes are activated, they will also release a myriad of pro-inflammatory cytokines, chemokines that will perpetuate and amplify the pain signal and the pathology that we are trying to treat. I just mentioned that earlier. I would say, from my perspective, what is interesting is that there is a balance between anti-inflammatory and pro-inflammatory cytokines, and depending which side this balance moves towards, the patient will recover or will develop chronic pathological pain. And you can see how an important aspect of this figure is these intracellular transcriptional cascades that take effect and lead to the activation of multiple chemokines that will, at the end, generate these different pro-inflammatory cytokines, what we call nowadays also these gliotransmitters from the neuron. So fundamental to this concept is that pain is the disease that we are treating. Imagine that you want to treat a patient with diabetes, and you observe that the patient is constantly going to the restroom. You decide then that this patient is losing too much water. And you decide that the best thing that you can do to prevent the disease is to restrain water. Unless you understand what is going on in these diabetic patients, and then unless you understand the role of insulin, what is going into the metabolism of glucose, you will probably have a very low chance to provide a appropriate treatment for the patient. Beyond whatever technical new developments that you have to first understand what are you treating, because that is going to determine how you're going to treat this condition. So here I'm trying to explain you how this pain goes from just an acute to a chronic, how it becomes in this transition. And there is amazing new experiments that are showing how, for example, animals that are exposed to a laparotomy, they do not develop any type of nociceptive or pain-like behavior. Similarly, if you have an animal in which you inject an endotoxin, LPS, you will observe that these animals do not develop any pain-like behavior. But if you apply the laparotomy first, and you wait several days, and then you do the LPS, you will observe a dramatic reduction in the tolerance of pressure of these animals. They are behaving like they have pain. What has happened? What seems to be happening is that you are priming, you are activating these glial cells in such a way that when you once again provide some form of injury, in this case endotoxin, the animal will respond with this marked increase in nociceptive-like behavior. If you apply medications, for example, like minocycline, at the time of the laparotomy, at the time of the initial injury, you will be able to blunt this response. But if you do it later on, you won't be able to do that, which means that this activation of the glial cells, if it could be prevented, could lead to not direct analgesia from these agents, but a normalization of the hemostasis in the central nervous system. We know nowadays also that neuroinflammation is the main mediator for these electrical signals to become chronic. And I think it's fundamental to remark that, because if you think about just the nerves, it's like a switch in your wall. You can turn on and off the electricity, so you will have light or not. If you want to observe something similar to what happens in pain, you need this switch to be somehow messed up in such a way that there is no control. And this is what the role of the neuroinflammation is in chronic pain. I mentioned earlier that if you want to do science, you have to start by an observation. And then probably the most important thing is, what are we stimulating? Isn't that a very honest question? I mean, we have been placing the leads in the spinal cord for 50 years in the posterior aspect of the epidural space, but without a clear understanding of what were we stimulating. So we should start by science. We should start by nature and the observation of what's going on. So we have done anatomical studies. We have observed the ratio of glial and neurons in the spinal cord. We have also observed what is the location and what is the quantity of these different cellular elements in different aspects of the spine. We have looked at the roles of these cells in chronic pain, and then we have been able to generate hypotheses out of which we can obtain results and further try to unravel these questions. This is the study that I mentioned earlier. What I think is remarkable, though, and again, this is monoclonal antibodies specific for each one of these different 4 types of cell populations. So we have for neurons, for astrocytes, oligodendrocytes, and microglia. And then what we did is we quantified that. This ratio of 12 glial cells per neuron happens in the dorsal aspect of the spinal cord. If you look at the dorsal horn, for example, the ratio is 20:1. That means that 92 to 94% of the cells underneath your lead or your active electrode are glial cells. And as far as I know, there is no magic electrical signal that can ricochet and go around these different cells and only stimulate the glial cells. And this is what I didn't mention in the last slide, but I think is worth to mention it now. This is what we observed. If you think about it, we observed that there was a marked increase in the number of oligodendrocytes around the middle of T9 and T9, T10, as well as the top of T10. What does it mean? How is it possible that you observe a higher concentration of these particular cells in a particular location, and what is the implication? You can also see how in the white matter, there is pretty consistent, maybe a slight increase around T9 of the levels and T9, T10 of the level of astrocytes. Now, just a little summary of what these cells do. I mentioned briefly earlier, the microglia are these immune cells that are in charge of sensing the environment, whether it's for a viral, bacterial infection, or for an injury. It can also detect if there is DNA that is abnormal or some damaged cells or something like this. It can sense and it can generate an inflammatory reaction based on that. Then we have the astrocytes, that probably the most fascinating aspect of these cells is that they do not only regulate the concentration of neurotransmitters at the synaptic cleft, is that the large size of these cells allows them to control the conduction of hundreds of thousands of synapses. And I don't know if you ever think about it, but with this, let's imagine that you are in a noisy place. You are maybe in a restaurant talking with a lot of people, and there is somebody close to you. Your ear is going to be able to specifically isolate the sound from this friend that is talking with you, so you will be able to maintain a conversation. How is it possible that our central nervous system handles all these thousands of different signals coming at different speeds? Let's imagine that you just hit your funny bone and you just feel this shocking pain at some point. think about it. All the fibers in this nerve have different depths. They will have different levels of myelinization. There will be sensory nerves. There will be motor nerves. And all this information is going to be triggered up into the brain. How does the brain coordinate these signals? And the answer is probably through the astrocytes. And finally, over the last 5 years or so, there has been a tremendous amount of research in terms of the oligodendrocytes, because oligodendrocytes, they can generate myelin for hundreds of surrounding axons, different from the periphery where a Schwann cell only covers one Rambier node or one section of myelin. Here, you have one cell that can control the speed of communication among hundreds of neurons simultaneously. I mentioned that I want to emphasize something that seems very simple. This is a study done in 1981. And you can see how the glial cells has a different resting membrane potential. In fact, the main intracellular ion is sodium instead of potassium, like in the neurons. So they have a totally different response when you are applying electrical signals. And I kind of hint what I'm going to mention right now is that, again, neurons are like males. It's an all or none phenomenon. Whether they have an action potential or they don't have an action potential, that's it. But when you look at the glial cells, they modulate so many different processes, from their concentration of neurotransmitters to the release of gliotransmitters to the release of different cytokines, chemokines, to the phosphorylation of multiple different ionotropic and metabotropic receptors. All of those clearly related to pain. And I mentioned also this early, just by modulating frequency, amplitude, or in this case, the way you recharge the balance. And this is an in vitro experiment in which they look at the release of glutamate when they apply a totally balanced, actively balanced signal, and you see no release of glutamate. But when you start going more and more into a totally monophasic stimulation, in this case, a cathodic stimulation, there is massive release of glutamate. I want to emphasize this once again. Astrocytes are not only large, but they communicate with each other through these gap channels that allow free flow of calcium waves along multiple cells, creating these islands of synapses that can control hundreds and hundreds of thousands of neuronal synaptic interactions. So why it is so important, the science? Why it is so important to have a hypothesis? Well, the main reason is we already observed that we were caged by the gate control theory for the last 50 years. And it formulates something that is totally true, the modulation of nociceptive signals by A-beta fibers. But the beauty of looking into this other concept is that it allows to formulate hypotheses, and the animal models allow us to explore a lot of different combinations of signals or independent variables to try to understand what is going on in terms of the moment that we apply electrical signals to our patients. This is the model that we are doing, and I briefly mentioned earlier that we are looking at transcriptomic expression. We are looking at RNA sequencing, which means that we can look at the whole nucleotide sequence of these animals. We are looking at around 31,000 genes. We are also looking by liquid chromatography and tandem mass spectrometry at the release of proteins. We have been able to identify around 7,200 proteins, and we also evaluate phosphoproteomics, which has allowed us to identify around 12,000 different phosphoproteomics of these different molecules. I already mentioned to you the idea of combining signals trying to target different components of biological processes engaged in pain was at the core of this hypothesis. we observe beneficial effects in animals in terms of behavior, for mechanical hypersensitivity, but also for heat and for cold. And based on that, a totally independent lab ran a research in which they use parameters that were follow those that we have used already in rodents. And you can see that this is pretty much the same type of response. They also observe a dramatic improvement, not only compared with the baseline animals that were not stimulated, but also when they compare with 1200 hertz, and in this case, 1000 hertz, 900 hertz, and 50 hertz. So this is the approach that we have been using. We have been looking at the molecular, at the behavioral response of these animals. But very early on, since 2004, we started looking initially with PCR at a small number of genes known to be related to pain. And then since then, we have transitioned to the current situation where we are evaluating the whole genome of the animals. And I would say even more than the genome, if you define genes as those that code for proteins, we are able now to evaluate the whole nucleotide sequencing, including what used to be called, but is wrongly so, the junk DNA. We can also look, so mRNA, and we can also look at proteins and phosphoproteomics. Our results in terms of upregulation of inflammatory response with conventional stimulation, as well as downregulation of ion channel regulation and synaptic transmission, was reproduced by the group at Johns Hopkins with June 1, where he observed similar patterns of upregulation and downregulation. Despite the two groups were using different models, we use an axotomy model, they use a neuroinflammatory model with the CCI, and our model was only 7 days, while their model was 39 days. So despite the differences in models and design, both studies concluded that spinal cord stimulation at low frequencies involved modulation of gene expression associated with immune and inflammatory response, as well as modulation of the synaptic transmission. And this is a study that we did in vitro. I was mentioning earlier I think was the same room, but I was mentioning earlier the hard time that we had to grow glial cells in vitro. This is some of the experiments that we did in vitro, and we look at a specific set of genes expressed by these astrocytes when they were exposed to electrical signals, in this case, different types of recharge balancing, whether it was a totally monophasic stimulation or it was a monophasic anodic or monophasic cathodic stimulation. And as you can see, these colors represent downregulation in red. Blue signifies that there is an upregulation. What I think is fascinating is that by simply changing the way you recharge your electrical signal, you will be able to modulate the expression of these genes that are relevant in immune function. Here we have, for example, GFAP that can be upregulated with asymmetric signals, or cathodic with passive recharge balance, but it can be downregulated with anodic signals. And those are some of the principles that we have been using when we try to design the differential target multiplex signal. And what is the importance of GFAP? GFAP is the gene involved in the production of GFAP, which modulates the release of calcium out of the astrocyte. We also look at genes involved in synaptic transmission, neuroprotection, something that I hope my lab is going to start looking more and more in depth. We have a lot of work into that, is these oxidative reduction reactions. If you think about it, it's quite obvious that when you are applying electrons and you are taking them out, you will be modulating some of these multiple, for example, ATP-related phenomenon to generate energy, and many other redox reactions, and also for adaptation of stress signals. So you can see, beyond what we already showed you by previous studies by Agnesi in 2010, that we were not only able to observe, like they did the glutamate, we also look at the expression of genes and how they were modulated by electrical signals. This is the same study, but now in vivo, and you can see in expression of different genes changes dramatically by changing the way you recharge your balance. This is kind of a dissection of the waveform. I mentioned earlier, for me, the waveform has passive, active, symmetric or asymmetric recharge balance. It has certain frequencies, pulse width, and amplitude. And the rest of what people call waveform is just a repetition of whatever initial signal they had. But what we observed was that there were fold changes in immune function-related genes, glial modulation, extracellular matrix, another aspect that is fascinating in my mind. And I think that, again, we have neglected all these proteins at the synaptic level in the extracellular matrix that also are known to lead to chronification of pain. We observed that there was a strong correlation coefficient in terms of this differential target multiplex for a set of specific genes that are differentially expressed by these different cell types. So we identified out of the 31,000 genes, we identified genes that were specific for each one of these cell populations, and only the DTMP compared with high rate or low rate had a strong correlation coefficient. Let me just point in the first column of the microglia. You can see how low rate doesn't try to reverse the expression of the signals towards the non-painful state. It basically further upregulates, because blue means, in this particular definition, it means that there is further upregulation of genes. And I wonder sometimes if the beneficial effects observed with conventional stimulation could be to the effects on the A-beta fibers, but this continuous pounding on the biological processes involving pain could somehow shadow the benefits of the modulation of the A-beta fibers. I was talking earlier about this important correlation between gene expression and proteins. We had a very high correlation coefficient. And also, I think it's interesting that some of the things that we can do nowadays, this is just a presentation in terms of GFAP, the gene that I previously mentioned that has always been of the astrocytic cells. And you can see how the GFAP gene and protein expression are increased in pain and are modulated towards the naive levels in the DTM program. What is fascinating nowadays is that we can look at phosphorylation, and I can tell you specifically at what particularly residue these particular proteins are phosphorylated in order to be activated or deactivated. And this is just, you have here the gene expression in blue, then in orange, the protein expression. And then when you use DTMP, you can see how there is a downregulation of those. And nowadays, you can even see where exactly this downregulation is happening. You see how the phosphorylated, more than even the gene expression by itself, but the phosphorylated proteins are dramatically downregulated by the use of DTMP. So people are claiming right now that, oh, we are also stimulating glial cells. And my answer to those that are claiming that is, yes, absolutely. Everybody has been doing that since the first time an electrical stimulator was put in a human body. The question is, as I mentioned earlier, low rate, for example, high rate, or DTMP, they have a totally different expression of genes. So what is the real result of these particular signals that have been applied to these glial and neurons? And are they normalizing the chronic pain towards the non-naive, non-painful state, or are they further upregulating or downregulated processes that were affected by pain? As I mentioned, other different controls had shown totally different expression in terms of genes. So the preclinical research is fundamental because it generates a working hypothesis of what we may happen to use different approaches, it may help to inspire approaches to be used in humans. And understanding the mechanism of action may help to improve the efficacy of the therapy. We have right now 12 months results of the randomized controlled trial showing superiority over conventional stimulation with this particular set of parameters. But I want to emphasize that by having this understanding, we can formulate multiple different approaches trying to provide the best relief in our patient. And it is the culmination of how an approach is inspired by preclinical research that can provide effective outcomes in humans. So what I will propose to finalize my presentation is that we change this approach that we have been following for so many years. Why don't we start from the bottom up? Why don't we start by the disease? What are we treating? So I will formulate that we need to understand what are we treating, what are we stimulating, and how these tissues at the neuroglial interaction respond to electrical signals. Only then we will be able to formulate specific parameters and we will be able to provide the best pain relief to our patients. Thank you very much. I have to stop sharing my screen. Okay. Is anybody there? Okay.
Yes, yes.
Okay.
Yeah,
I was saying that I was talking to myself.
Thank you. It was wonderful, my friend, and I have no words to say what it was your interpretation. So very good, my friend, very good, and you can understand very things about the mechanisms and the different ways of spinal cord stimulation. So I'd like to call my friend Tiago and Chris, if you want to introduce my friend Tiago to final presentation, and after Tiago we have the final discussion, okay?
Leandro, vocês estão vendo a minha tela?
Ainda não.
Fala, Tiagão. Tudo bem, meu amigo?
Fala, chefe. Estou tentando compartilhar aqui.
Tiagão, não é fácil, mas a gente chega lá.
Você está vendo o maisinho ali da esquerda, Tiago, em cima?
Eu cliquei nesse símbolo aí.
Mas você estava com a tela do PowerPoint aberta antes? A sua apresentação estava aberta? Ou, sei lá,
Não, eu minimizei ela. Aí eu apertei aqui, compartilhar a tela.
Bota a tua apresentação no background do computador, tá?
Uhum.
Aí depois você faz o compartilhamento da tela, tá?
Então, deixa eu tentar aqui. Compartilhar a tela. Carregando tela. Aí abrir o menu compartilhado, é isso?
Aí você aperta no maisinho, né?
Aí eu cliquei aqui, compartilhar a tela.
Maisinho.
Sandra, será que ele não está com o mesmo problema do Vallejo?
Pode ser. Tiago, você foi no maisinho lá e abriu a lista, o menu?
Abri, está escrito aqui, compartilhar a tela. Aí eu já
Isso, foi lá.
cliquei no compartilhar a tela. Aí aparece carregando tela, escreve itens compartilhados serão exibidos aqui.
Tá. Faz o seguinte. Tu tá no Google Chrome?
Estou no Chrome.
Tá, e você está no Mac? No Mac, Tiago?
Mac.
Tá, então vai lá em preferências do sistema.
Meu Deus.
Vamos lá, não é rápido.
O Vallejo vai ensinar agora ele. Vamos lá.
Segurança e privacidade.
Segurança, privacidade, meu Deus. Ah, tá aqui.
Aí, à direita. Aí. E aí você vai nesse menu da esquerda até gravação de tela.
Gravação de tela?
Vai descendo.
Aqui só tem
Screen record? Não?
firewall, privacidade, serviço de localização. Não tem isso aqui não, cara.
Não, tem, ó. Você vai descendo. Ele começa assim, ó. Serviço de operação, contato, calendário, vai descendo.
Calendário, lembretes, acessibilidade, diagnóstico e uso. Só isso.
Vai descendo que tem mais coisa. Vai descendo.
Não tem, velho. Tô descendo aqui.
Gravação de tela.
Não tem, não. Não tem.
Não.
Vamos fazer o seguinte.
Tela inteira.
Opa. Ô, Tiago, sai e entra de novo.
Quer?
Sai e entra de novo para ver.
Tá bom. Então, tá.
Pode ser.
Já estamos aqui com o Dr. Caporal. O André também está aqui, né, André? Bem-vindo aí, viu?
Vou sair, então. Vou sair e volto.
O Caporal está aí? Caporal está aqui. Tá. Vamos ver. É uma dificuldade a gente ter que realmente ver isso aí amanhã. Tudo bom?
Fala, Cris. Tudo bem, Cris?
E aí, Cris?
Tudo certo?
Vamos ficar todo mundo para a discussão, hein? Que vai ser a parte boa, hein?
Vamos ferver aí, pessoal. Vai ser boa a discussão.
E se ele entrou aí de novo?
Tá aqui?
Fala, Tiago. Tenta novamente agora.
Tô tentando de novo. Compartilhar a tela, não vai.
Não tá carregando.
Ô, Thiago, é grande a apresentação?
Não, muito grande, não.
Eu quero mandar no meu zap aí, daí eu já compartilho aqui, tá ok?
Posso mandar pro seu e-mail, né?
Zap o e-mail, isso aí.
Manda aqui, enquanto vai tentando aí, eu projeto aqui.
Putz. Vamos ver aqui.
Vai ter que ver isso aí. Dr. Leo.
Yes.
It's a pleasure. Welcome aboard. we are having some difficulties with the system, but just a few minutes for the presentation of Dr. Tiago, and after that we have the final discussions. Is it okay for you?
Yeah, that's fine.
It's a great pleasure to have with us. It's a great honor for us. Yeah.
Thank you very much. I appreciate it.
Tô mandando aqui. Putz, cara, tem algum... Esse negócio...
O pessoal não ajuda, não, Zé?
Deixa eu tentar ver aqui.
Tá com application. Jonas, te mandei.
No e-mail?
E-mail.
Beleza.
A gente consegue abrir. Tá de novo.
Tenta aí de novo, Tiagão.
Começa a carregar e não há itens ativos.
É lá no negócio do sistema, Tiago. Engraçado se não abrir aquele negócio lá da...
Não, abri, mas não tem aquela opção.
Você tá com o celular aí na mão?
Tô. Mandei no e-mail.
Faz o... Abrir aqui, ó. Chegou aqui para mim. Tá abrindo. Apareceu aí?
Opa,
Apareceu.
Deu certo aí?
deu certo.
Ok, então vamos embora. Você vai passar?
Sim, só falar. Oi.
Vamos lá. Gostaria de agradecer o convite, né, de estar nessa aula e com esses gigantes aí da neuromodulação, Dr. Caporal e Dr. Vallejo. Tá? Eu não tenho conflito de interesse nessa apresentação. Alguns slides foram cedidos pelo próprio Dr. Caporal e o Dr. Bernardo Mônico, alguns também. Bom, vamos falar primeiro da vida real, né? O que a gente tem de indicação para estimulação? E Jonas? Próximo, por favor. Bom, tem 4 doenças que têm uma evidência principal aí na literatura que a gente pode usar a estimulação, né? Nós temos a Freiberg Syndrome, né? A síndrome pós-laminectomia, a angina refratária, a isquemia periférica de nervos, né, de membros, que muito pouco utilizada no Brasil, e a síndrome do complexo regional tipo 1 e tipo 2. Então, esses são os pacientes que têm a maior chance de responder com a neuromodulação hoje na literatura, né? Pode clicar, próximo, por favor. Mas a gente tem outras doenças, né? Neuropatias periféricas diabéticas, injúrias faciais do plexo braquial, dor perineal, radiculopatias, mielopatias, dores lombares, né, de uma forma geral. Tá? Pode passar o próximo. E a gente tem um dos consensos da ANS, da Sociedade Internacional de Neuromodulação, ele fala sobre essas evidências e recomendações. Próximo, por favor. Pode passar mais um. Nós temos para síndrome pós-laminectomia, evidência grau 1 na literatura e evidência A do consenso. Nós temos para dor de extremidade, evidência 1, 2, né? E para síndrome do complexo regional também, evidência 1 e recomendação tipo A. E falando das formas de onda, pode passar. Né? Então, isso a gente vai cair nas evidências. Agora vai o outro lado da aula. O que a gente tem de opções? Nós temos estimulação tônica, que até 2010 era a única forma de estimulação que a gente tinha. Temos burst, ou estimulações salvas. Temos high frequency, que é o HF10. Temos o high density. Temos closed loop stimulation e temos o DTM. Então, nós temos 6 opções de formas de onda, né? E o que a gente tem na evidência científica dessas formas de onda? A gente tem estimulação tônica, nós temos resultados na literatura, com melhora, por exemplo, para a síndrome pós-laminectomia. Não dá aqui pelo tempo para falar de todas aquelas 4 principais, mas vamos pegar a síndrome pós-laminectomia, que é a que tem são o maior número de pacientes que a gente vai usar. Então, nós temos vários trabalhos na literatura com longo follow-up, né, que mostra que estimulação tônica funciona. Tem melhora de qualidade de vida. Pode passar o próximo, Jonas. Satisfação dos pacientes, né? Então, estimulação tônica funciona. Mas a gente tem aquele paradigma da estimulação tônica que o Dr. Vallejo falou, o Dr. Golovac falou na aula dele. O que a gente fala para o paciente é 50% de chance de melhorar 50% a longo prazo. Então, esse é o paradigma da estimulação tônica que a gente vai comentar mais para frente. Nós temos burst. Burst tem resultado na literatura que funciona. Teoricamente, é uma onda mais fisiológica. É uma onda de capacidade, teoricamente, de mexer com a via medial da dor, mexer com os componentes afetivos da dor. Nós temos várias, né, desde 2010, quando as primeiras publicações de Reader, né, até hoje, até o estudo Sunburst, que é um estudo que tem mais de 1.000 pacientes no mundo todo, né, que reuniu uma compilação desses pacientes, nós temos resultados importantes, principalmente quando você compara a estimulação em burst, né, com a estimulação tônica. Nós temos melhora na dor lombar, melhora na dor nos membros e melhora da dor em geral. Pode passar o próximo. E o high frequency, né, começou a partir de 2013, o primeiro estudo foi de TID, né, é um novo conceito, é uma nova frequência, uma nova largura de pulso, um novo conceito de implante anatômico. Pode passar a próxima. Nós temos excelentes trials, né, conduzidos, né, acho que parou o compartilhamento aí. Nós temos excelentes trials, né, conduzidos pelo Dr. Leonardo Caporal, está aqui. Nós temos o Ciência europeu e o Ciência americano, né, que mostra, né, para a dor lombar em membros inferiores uma comparação com a estimulação tônica brutal. Né? Você tem a longo prazo, no follow-up de 24 meses, pacientes que mantiveram mais de 75% de melhora, né, na dor, agora está de volta aí. E enquanto que na estimulação tônica, Pode dar mais um clique aí, Jonas, por favor. Quando você compara com os estudos mais bem conduzidos a longo prazo, que foi do Kummer, do North, você tem uma melhora inicial, né, nos testes de quase de 80% para mais, e você tem uma queda nesses índices a longo prazo, né, uma habituação, né, que é um dos problemas da estimulação tônica. Pode passar para o próximo. E esses estudos mostraram, comparando com outros estudos de estimulação tônica, né, com a Boston, com outros estudos, inclusive com o Sunburst, uma melhora nas escalas de dor interessante, né, maior ainda do que a gente viu. Pode passar o próximo. Para High Density Simulation, a gente tem muito pouca evidência, né, isso foi uma resposta, né, de uma companhia, a estimulação não parestésica. O que a gente tem de estudos são alguns pacientes para tentar salvar, né, aqueles pacientes com estimulação tônica que têm habituação. Então, já a High Density não tem tanta evidência científica. Pode passar. O próximo. E nós temos ainda os ECAPs, né, são Closed Loop Simulation, feito por uma companhia australiana, a Saluda, que, pode passar o próximo, desenhou 3 estudos bastante interessantes, o estudo Panorama, que foi o inicial, depois o Avalon, que foi multicêntrico, né, com N maior e com 5 centros na Austrália, e por último o Evoke, foi um estudo randomizado, controlado, duplo cego, com 134 pacientes, né, pode passar o próximo. E o Avalon já tinha mostrado os resultados muito interessantes, né, você tem 70% dos pacientes com mais de 80% de melhora da dor lombar em 3 meses, o segmento é mais curto, não é tão longo quanto foi o estudo feito pela High Frequency, 80% desses pacientes mantiveram esse benefício com 6 meses, né, e 8 de cada 10 pacientes tiveram melhora no sono, ou seja, melhora da qualidade de vida. Pode passar o próximo. E tem um estudo multicêntrico deles, que é o Evoke, pode passar, por favor, que mostrou vários resultados interessantes, né, em 12 meses, 80% de melhora na dor lombar. Próximo. 56% dos pacientes altamente respondedores, né, mais de 80% de melhora. Próximo. E melhoras em várias qualidades, várias escalas de qualidade de vida. Próximo. E nós temos DTM, o Dr. Vallejo já comentou, né, uma terapia diferente, né, uma estimulação de glia, tem uma forma particular de programação. Pode passar o próximo. Próximo, por favor. Mais um, está voltando. E tem esse estudo que saiu recente, né, com um follow-up de 12 meses, né, mostrando uma melhora. Pode voltar, Jonas. 2 slides, por favor, cara. Uma melhora significativa, né, na resposta, né, desses pacientes, 84% de melhora de dor lombar, né, nesse estudo. Pode passar o próximo.
Eu estou mandando ele faz isso com animação.
Vai mais um. Aí. Então, falando de evidência científica, o que a gente tem? Closed Loop Stimulation, High Frequency Stimulation, DTM, principalmente esses 3, tem uma evidência alta de melhora. Pacientes, eles tiveram melhora nas escalas de dor de 80% ou mais. Burst também, e todos parecem ser melhores do que estimulação tônica na literatura. Burst tem mais papers falando de recuperação de pacientes que tiveram tolerância, né? E High Density Stimulation, realmente, a evidência é muito baixa. Próximo. E como é que é a vida real, né? Que é a parte de cá desse tema dessa aula. Então, evidência científica versus vida real. Então, o que a gente tem no Brasil? A gente tem estimulação tônica, a gente tem burst, a gente tem high density, a gente tem DTM. Nós não temos high frequency, né, 10.000 Hz ou 10 kHz, infelizmente, e não temos ainda closed loop stimulation. Né? Próximo. E a gente tem que pensar sobre tolerância, sobre habituação. Tá? Então, para pacientes virgens, baseado na evidência científica, ou seja, aqueles pacientes que vão fazer o primeiro implante, eu tento usar estimulações não parestésicas. Né? Eu ainda faço teste para implante, eu acho importante por questões econômicas no nosso país. Tem vários artigos mostrando que isso não é tão relevante, então é muito discutível, não é o tema dessa aula. Mas nesse mês chegou o Invisible Trial, que é o teste em estimulações salvas, e eu acho que vai ser interessante testar. E você pode fazer DTM de teste também, se eu não me engano, no novo sistema que foi liberado pela Medtronic. Então, para pacientes virgens, eu tento estimulação não parestésica. Lembrando que estimulação tônica funciona, e funciona muito bem, e tem pacientes que vão preferir estimulação tônica. Na minha casuística, eu vou dizer para vocês que 30% dos meus pacientes preferem estimulação tônica do que a estimulação não parestésica. A maioria ainda prefere não parestésica. Isso pacientes virgens. E a gente tem que pensar sobre tolerância. Por que a gente tem que pensar sobre tolerância? Próximo. Próximo, por favor. Porque tolerância é a principal causa de falência da terapia. Existem vários estudos com uma alta quantidade de pacientes, por exemplo, esse estudo do Salim Hayek, 7 anos, que a maior causa do explante nesses pacientes foi perda do efeito terapêutico, ou habituação, ou tolerância. Próximo. Tem um outro estudo interessante que vai aparecer aí, de 22 anos de follow-up, 410 pacientes, e que a principal causa também de 85 explantes desses 410 pacientes foi tolerância. E por que isso acontece? A gente ainda não sabe, a gente acha que é uma modulação do sistema nervoso, ele vai acostumando com o estímulo medular, e se habitua. E aí eu trouxe 2 casos para a gente discutir que evoluíram com tolerância e que a gente conseguiu fazer o resgate. Então, primeiro é um paciente, um homem de 54 anos, com história de cirurgia prévia, por uma dor teoricamente discogênica, e parecia ser mesmo, há 3 anos atrás, antes de vir no consultório, tem uma artrodese L4, L5, S1, que eu não coloquei aí, e que vem evoluindo com uma dor caracteristicamente neuropática, uma dor tipo choque, irradiando para o membro inferior direito, os dermatos L4, L5, S1, tem parestesias no trajeto, EVA de 9, limitante, qualidade de vida. No exame físico ele tinha uma hiperpatia ou hiperestesia nesse membro, nesses dermátomos, fez tratamentos prévios com fisioterapia, acupuntura, utilizou moduladores de dor. Ele é um paciente que tinha uma condição econômica ruim, não conseguia nem comprar, por exemplo, duloxetina, um paciente que tinha um convênio mantido ali pela empresa que ele trabalhava, chegou a altas doses de gabapentina, iniciou tramadol sem resposta e estava usando já morfina 10 mg de 4 em 4 horas, com 2.400 de gabapentina. Próximo. Próximo, por favor. O que a gente fez primeiro? Investigar a instabilidade, foi o que o Vallejo falou, naquele estudo dele de neuromodulação, ele excluiu pacientes com instabilidade. Pode passar, por favor, Jonas? Mais 3 cliques aí. Então, esses são os raios-x desse paciente, em flexão e extensão, esses são as tomografias, a gente não achou nenhuma instabilidade. Pode passar. Passa o próximo, please. Mais um. E a gente começou com procedimentos diagnósticos minimamente invasivos. Neuromodulação não foi a primeira opção. A gente fez bloqueio das raízes mais simpático. Colocamos uma bomba elastomérica. O paciente ficou com melhora mais de 80% da dor por quase 30 dias. A gente repetiu esse procedimento, fez pulsada nas raízes. Não melhorou o tempo de resposta. Fez um epidurograma. Quando a gente fez esses bloqueios, ele tinha uma falha de enchimento. A gente fez epiduroplastia, ele também não melhorou. Próximo. Próximo. Então, fizemos o teste com eletrodo, estimulação tônica, o paciente melhorou mais de 90% do EVA, cobriu a área de dor, e a gente fez um implante permanente. Então, ele ficou 10 meses, chegava no consultório, ele até ajoelhava pra gente. E depois ele voltou a ter dor neuropática no mesmo padrão, mas ela ficou ali na região lombar. E o que a gente faz com o paciente com habituação? Próximo. Tentou um bloqueio na sacrilíaca, ele não teve resposta, revisamos o eletrodo, não tinha mudança, e a gente acabou tentando uma terapia de resgate. Próximo. Próximo. Então, esse paciente, volta, por favor. Esse paciente, Vallejo, ele tinha um implante Medtronic. A gente tinha 2 opções no Brasil, na vida real. Ou a gente trocava por um implante que faz estimulações salvas, pode ser da Boston ou da Abbott, que são as 3 marcas que a gente tem, ou a gente trocava por um Medtronic, da mesma marca. E o DTM já tinha sido liberado. E a gente fez uma estimulação com DTM e a gente teve resgate da terapia desse paciente com DTM. Foi um caso muito prazeroso, porque o DTM não foi ainda, teoricamente, a indicação dele não é para resgate de terapia. O paciente pós-laminectomia e com dor lombar e comparado com estímulo tônico, tá? Então, esse foi um caso de vida real, que a gente teve a sorte de resgatar com DTM. E o último caso, para terminar a nossa aula, um caso que eu até já apresentei num dos congressos da Sobramid, é uma paciente jovem, sexo feminino, que fez uma cirurgia de varizes, e depois do procedimento da anestesia espinhal, ela sentiu um choque no trajeto dos membros inferiores e ficou com uma dor de característica neuropática, mais perfeitamente uma síndrome de dor complexa regional. Próximo. Ela veio ao consultório, estava extremamente revoltada com a situação, né? Ela foi fazer uma cirurgia de varizes, ganhou uma dor neuropática, uma síndrome de dor complexa regional, uma dor limitante e incapacitante. Próximo. A gente tentou os tratamentos conservadores para ela, né? Fizemos a avaliação radiológica e tentamos tratamentos conservadores, assim como a gente fez para outro caso. Próximo. Essa é a ressonância dela. A gente não vê tantas alterações estruturais. Essa é a eletroneuromiografia mostrando a radiculopatia. Próximo. E não melhorou, né? A gente tentou todo o tratamento conservador. Partimos para procedimentos de bloqueio. Bloqueio simpático lombar, ela teve uma boa resposta, mais de 70% de melhora. Foi transitório. Fizemos uma sequência de bloqueios simpáticos, que também não durou muito. Próximo. Depois fizemos uma radiofrequência, durou 6 meses. E posteriormente, em 2014, isso é um caso antigo, a gente discutiu estimulação para ela. E a gente fez um implante cirúrgico. Tá? Próximo. Na época, em 2014, a gente não tinha nenhuma dessas formas não parestésicas ainda disponíveis aqui no Brasil. Então, a gente implantou um eletrodo em placa, um 2x8, da Abbott, cobriu. Próximo, por favor, Jonas. O paciente teve uma excelente melhora, uma cobertura da área de dor. E aí, 2 anos de lua de mel. 2 anos depois, tolerância de novo. Mesmo caso, né? O que a gente fez? Avaliamos impedância de eletrodo, tentamos novas frequências do que dava para fazer naquele programa. Washout, né? Tentamos desligar o eletrodo para o paciente desabituado a estímulo tônico, depois ligar de novo. Bloqueio simpático venoso, não tivemos, né, nenhuma resposta. Próximo. Então, o que a gente discutiu? Em 2017, né, ela começou a habituação em 2016, em 2017, vamos trocar essa bateria, esse gerador, e a gente preferiu uma estimulação não parestésica. E colocamos um burst e ela teve uma excelente melhora do quadro de dor, mais de 70%. Ela resgatou aquele benefício que ela tinha no começo. E mais impressionante, a gente observou uma melhora do humor. O paciente realmente teve uma mudança de personalidade. É lógico que com a melhora da dor isso vem, né? Mas é uma paciente que foi muito gritante, né, a alteração que ela teve no comportamento. Próximo. O paciente estava estável. Depois de 1 ano, ela teve esgotamento da bateria. Um gerador não recarregável, né? Próximo. E aí a gente pensou, vamos pôr um gerador recarregável, né, que é o PROD da Abbott, mas o PROD não é aprovado para ressonância de corpo inteiro. É um paciente jovem. E aí a gente resolveu testar de novo o não recarregável. Próximo, Jonas. Próximo, por favor. Aqui está falando das vantagens e desvantagens. Eu não vou me alongar, a aula já está terminando. Próximo. E aí, esses são os parâmetros que ela usava. E aí a gente colocou o não recarregável. Volta 1. E aí fizemos o protocolo BOLD, né, que é o protocolo novo de estímulo, que o paciente não fica o tempo inteiro fazendo o estímulo, né? E é o que o professor Manuel comentou, né? Talvez seja mais fisiológico para a medula utilizar uma estimulação com pausas, né? Próximo. E essa paciente ficou bem até hoje, ela está com a mesma bateria, desde 2018 que a gente trocou. Então, esses foram 2 casos para mostrar o que a gente tem de opção no nosso país, na vida real, para evitar a tolerância. Então, as conclusões, nos últimos 10 anos, a gente tem uma revolução nas formas de ondas de estimulação medular. Nós temos mais de 6 tipos diferentes, né? A gente tem evidências científicas na literatura que closed loop, high frequency, DTM, tem até mais de 80% nas melhores escalas de dor lombar e membros inferiores desses pacientes. O burst também mostra superioridade ao tônico nos estudos. As estimulações não parestésicas parecem ser melhores para evitar a tolerância. E a gente tem que levar em conta algumas coisas, né? Experiência pessoal, experiência de programação. E no nosso país, eu acho que é isso que a gente tem de armas por enquanto. A gente espera que o resto chegue logo. Pode passar o último, por favor. Muito obrigado. Queria agradecer o convite e de estar aqui com esses gigantes da neuromodulação, Heitor Vallejo e Dr. Leonardo, para discussão. O último, Jonas. Mais um. Lembrando o nosso curso de neuromodulação invasiva, para quem tiver interesse, no ano que vem as datas já estão aí. Tá, e um curso endossado pela ANS. Obrigado.
A gente que agradece, Tiago, pela palestra. E, pessoal, o curso do Tiago realmente é fantástico. Se tiverem a oportunidade de fazer, façam. E eu queria abrir, então, para discussão. Né, Jonas? Fantástico o curso. Queria abrir para discussão, então.
Thank you, Tiago. We are speaking English because Dr. Leo is with us, and wonderful presentation. And for us, it's a great honor for the speakers here. And for me, it's like a dream. Be with you and my partners, Dr. José Luis and Dr. Alexandre Mil. I have to thank you and for you, for everybody, all the speakers, and to be here in this time. I know the difficulties and the time that we are living. So thank you very much to all. Okay.
Thank you.
Thank you. Thank you very much.
Thank you.
Thank you, guys. Maybe it's better to start the discussion. Yes, thank you for coming.
Names of neuromodulation, I have to. I don't want to talk too much because the names that we are here, Professor Doni Manuel, Dr. Alexandre Amaral, and the other speakers are the leaders in the world. So you can talk about that.
I have a comment, a question. May I? To Dr. Vallejo. And about the magnetic stimulation and the neurotrophic power of a magnetic stimulation instead of electricity.
Yeah, I mean, in theory, anything that creates a change in the electrical field can work. I mean, and again, my previous conversation was without wine. Now I have wine on top of me, so that would probably provide totally different ideas. If you think about it, a neuron, a glial cell, all of them have. If you think about quantum mechanics, you have all these molecules moving around, and anything that can magnetize these electrical charges of those cells can modulate the condition and can modulate the biological processes that they run. So it's absolutely rational to consider that electrical lasers, for example, light can modulate this modulation or constant movement of particles. The same will happen with sound systems. There is so many different ways that you can apply energy. At the end of the day, is the application of energy and the movement of electrons from one side to the other between the different components of the system that will lead to the response. We have been using the electrical signals because it's easier. We did some initial experiments with lasers. They will definitely work, but the challenge will be to apply the correct waveform without creating heat, and whether it's invasive or not. And I can tell you also, I had to learn a lot about business. That was never my goal, but when I started running Steamgenics and the study. And one of the things that is particularly challenging is if you want to create something totally novel. So if you come tomorrow with the idea to implant a new system with sound waves into the epidural space, you're going to face dramatic restrictions from the FDA in the United States. You will need to do a lot of safety studies, very long-term studies, and I'm 58, so I have to be careful of what do I get into. But absolutely, the answer is yes, those things can be pursued.
Thank you.
We have some ongoing electromagnetic field studies right now. I'm doing one on peripheral nerves with a small company called Neuromodulação, which is in Los Angeles, and it's ongoing right now. The device is very elegant and provides profound relief of the pain. However, affects the single peripheral nerves. So indication is post-surgical mononeuropathy, like ilioinguinal genitofemoral neuralgia, saphenous damage of the nerve, and so on and so forth. For now, what I could see from the pilot, it works really well. And if you think about it, this type of certain type of waveforms with a high electromagnetic field can be used very early in the algorithm of the treatment of such pain. So, and if you think about gabapentin and pregabalin, the pyramid, sodium channel blockers, and others that we use as a membrane stabilizer is the first line of the therapy, we have a major problem with them. Look at the Lancet study back in 2015 showing how many patients we have to treat to get one with more than 50% of the pain relief. For gabapentin, which is the best tolerated, is 7.2. 7.2 patients for one. That actually improves with more than 50% of the pain relief. So we have actually much better tools. I'm not talking about powerful tools like DTM or 10 kilohertz or something. But for example, using this type of modified electromagnetic fields as a non-invasive therapy in mononeuropathies can change totally the algorithm of the treatment in a favor of this type of the therapies before you even consider putting somebody on gabapentin.
Dr. Leo, the number needed to treat for pain neuroscience education, just education, for function is 2. For pain is 3. Is better than pregabalin and gabapentin. So these drugs, they can act only for a few hours, and it's much better to try to undo the neuroplastic adaptive changes. And I guess I think that burst, high frequency, education, all these kinds of approaches, they can modify, they can modulate how pain is perceived by patients. Much better than drugs.
Yeah, I think that we have to insist on certain changes in the algorithms in the future, because we have too many steps initially that are applied by primary care physicians and others that are really not working well for our patients. We see so many patients that are frequently depressed on gabapentin. We see the patients that are walking in the cloud all day and stuff like that. So I'm just talking about some of these initial steps that may be changed, like Dr. Jacobson said about the non-invasive therapies.
Dr. Vallejo, you have a question from Dr. Newville. What are your idea, because DTM does not cause this fatigue of stimulus as tonic stimulation?
First of all, thank you, Nilvio. It's a pleasure to have this question from you. I would like to make a little comment about fatigue or tolerance, as it has been discussed today.
is, first of all, if you talk about the BOLD study, the BOLD study had originally 52 patients, which were, the way the study was conducted is the patients were exposed to a very significant reduction in energy consumption for a very obvious reason, which is what Tiago exposed in his presentation, and is that this high frequency 500 hertz, I'm sorry, which is what is used in the DPM in the burst DR, you will obtain a very exhausted battery very early on. So what the BOLD study was designed for was to try to obtain parameters that could save battery and avoid these early replacements. So if you look at in detail, like everything in the world, if you look in detail, you will observe that there is 52 patients originally that were trialed, but out of those, only 22 end up in the arm with this 30 seconds on, 6 minutes off. And then if you look at 6 months, there is only 12 patients that did respond to this particular paradigm, which means that in total, the response of this therapy is less than 20%. So this is something to consider. Now, it's also important to consider when you talk, for example, about the Sunburst study. The Sunburst study had a total of 39% of the patients with more than 50% relief. So it's 39% in the active arm, the burst DR, versus 32% in the control arm. So those are some numbers that are out there, they are published, and I think is good to consider. But what I would say pertinent to the question that Nilvio is asking is, first of all, why are they formulating these 30 minutes on, 6 minutes off? And it's very obvious. It's not because there is a good rationale, it's because the battery is burning. So that is bottom line. So they need to create something that hopefully will work, but there is no rationale. So the concept of tolerance, I will argue, has to be considered in a more scientific way. I will argue that if you have a patient with tonic stimulation, and I had those patients, and I'm sure you all had, that have been implanted for 3-4 years and has no relief whatsoever, if you ask them, are you feeling paresthesias? The typical answer will be, yes, I'm feeling paresthesias. And then you can play, like you did, Tiago, with your patient, you move cathodes, anodes, you try multiple different programming. At the end of the day, it doesn't work. Is that tolerance? I would say no if you think about the gate control theory. Because if you think about the gate control theory, the main goal is to obtain paresthesias. And the cells are not tolerant because the cells are still providing paresthesias. So there is no tolerance at the level of the neurons. What I try to present, though, in some of my preclinical research is that some other different pathways, molecular pathways, can be modulated in a way that they can end up affecting this pain in these patients. So it was very clear, and we demonstrated that. June 1 did the same type of study, that when you apply certain parameters, you can be further activating biological processes that are detrimental in pain. Now, the question that I think is fascinating is, can you find out what biological processes are benefited with your particular waveform? It doesn't matter which one. And then if you can determine, okay, it takes me 3 hours or 2 hours, and I'm talking more in terms of half an hour, hours. I think the 30 seconds is just a very desperate attempt to save battery more than a scientific attempt. I don't think that you will generate transcriptional factor changes in only 30 seconds. But if you think from a rational perspective, I think it makes a lot of sense that you may want to determine how long does it take for you to reverse certain transcripts. And once you do that, then you can turn off the signal. For how long? I don't know. I would say it will depend on how long does it take for these biological processes to be reactivated. But in theory, you can create a rational approach in which you will be turning on and off the signals depending on how long does it take for biological processes to be modulated. And that makes a lot more sense when you are talking, for example, about this biological processes that are modulated by paresthesia-free stimulation paradigms. Now, the other thing that I think is very important to consider is this medial pathway, because when you talk about the medial pathway, there is at least 2 studies that I'm aware of. One of them is with functional MRI by Dr. Ali Resai, in which he demonstrated that conventional stimulation activates medial pathway. And if you look at the studies originally done with EEG for medial pathway, you will observe that the first study included 15 patients, but only 5 had the EEG. Why 5 and not the 15? And which one of the 15, the ones that responded or the ones that didn't respond? What is fascinating is that the same 5 patients were published in a second article, exactly the same 5 patients. So I would say, and in fact, I learned recently that the reader had accepted that, for example, high-frequency stimulation could also be modulating medial pathway. So that means, in other words, that it may work. I'm not at all disagreeing with the response, the clinical response. What I'm saying is, first of all, there is not a mechanism of action. The fact that you say, oh, I activate the medial pathway resembles, that's a beautiful baby. It resembles this label that was used in the movie The Time Machine. You just, okay, what are you doing? Oh, I'm moving the medial pathway. How? How do you do that? I mean, how do you get up there? Because there has to be a process. So what I'm insisting that we all have to do is to be more scientific and go back and ask to people, why are you doing that? Tell me the reason. Show me the money, because it's very easy to talk in the podium, especially when you have 10 minutes and just show up like a marketing strategy. Hey, we are good because of this and this and that. Okay, calm down. Show me the bold studies, show me the results, show me these other studies, and then show me why should I expect this or that. Saying that, answering to Neilville, would my system prevent tolerance? I don't know. That is the answer. But we are trying to have a rationale to hopefully one day avoid these effects from a scientific perspective, not from a marketing perspective.
Perfectly, and you have to choose the patients. It's a question. It's what patients you have the kind of modulation and the kind of waveforms, and which patient and which waveform we choose for each patient. It's not a recept. So when you prescribe electricity, and what kind of electricity and we should do for each patient, for that's it's the big difficulty that we have. Yes.
I don't know if I think Leo will probably have also something to say about it. I'm a total advocate for randomized controlled trials. I think this is the top of the chain in terms of research. So I would say high frequency stimulation has clearly demonstrated superiority over conventional stimulation, no doubt about that. I also think that if you look at this particular study, you clearly understand that 86% of the patients in the Sensa trial had axial low back pain, and these patients responded in the round of 80%. So that is a very clear group that I will use for that. The second group that I will definitely think is, I think DPM, but still the results need to be published in a peer-reviewed journal, and we are working on that. I think that that also will bring more evidence into the field. If you think, again, at the sunburst showed superiority over conventional. For what pain? I don't know. That's a good question in my mind, because the patients are happy, but are they happy because they have less back pain, or because they have less leg pain, or because they just feel happy? If you look at the study carefully, you will not have the answer that you are looking, Leandro, because if you are telling me, I have a patient that comes tomorrow with axial low back pain, which one will I select? I don't know. It seems like burst is better than conventional, but I don't know for what, based on their own study. It doesn't mean that in the real world, it doesn't work and it's fantastic. I'm just saying, if you base your determination on the evidence that is published, then you will have some trouble. Patients with CRPS, I think right now the DRG stimulation is probably the one that has shown better results with the accurate study. I will prevent people, though, especially when they are starting doing these procedures, it is very challenging. Placement of the lead is challenging, and getting the lead into the foramen could be a challenge. And there is two things that Benjamin Franklin said that were short in life. One is death, and the other one is taxes. I will add a third one, which is foraminal stenosis. Just wait, and eventually you will develop foraminal stenosis. If you put a lead there, I know putting them is a challenge. Removing them is another big challenge. So I think that, again, but saying that, that's the best evidence that we have for CRPS. I don't know, Leo, what is your opinion?
Yeah, I mean, I can just say that we kind of progressed from what Nagy Mikhail was calling 50-50 club, 50% of the patients with more than 50% of the pain relief. I still believe that what you call high density and low frequency, all of those from 0 to 1500 or 3000 hertz therapies belong to the same bucket. And then you have this distinct three therapies with the randomized prospective studies, and I brought that in my talk earlier on, that belong to 80% club. And that's DTM, that's closed loop type of the stimulation, and that's high frequency. Let's see if we're going to have level 1 evidence for anything else. But we don't have right now. We don't. I mean, we do like this type of the competition. We do like this type of the, what Leandro mentioned, mess, because now we have so many therapies and we wonder what we're going to pick. But this is actually good for us as interventional pain physicians, because I can see now more and more folks investing into the new therapies. There's several new players. We just finished a big randomized prospective study. I was fortunate to lead that with Biotronic. They have also a new type of the waveform, which looks really good. 500,000 hertz, I mentioned this before. We're initiating now RCT. It looks really good. But for now, the present evidence with the large RCTs suggests that those three therapies stand out from anything else. And, I mentioned also before in cardiology, if you have a RCT that shows that 80% of the patients survive with a new stent, everyone ditches this 50% stent out because the human life depends on it. Same in oncology and so on. In chronic pain, we are sort of like, it's okay, whatever. It's not. I think that the evidence matters, and personal experience or second wave of the RCTs, especially initiated from academic institutions, should prove that. We have to have, like in cardiology, for every therapy that we use, at least 2 randomized prospective trials. And then we will be able to say, okay, the evidence is there. I feel comfortable that my patients are getting the greatest benefit that they can. I think we are on that way. I think we're not going to have this discussion probably in 5 years from now. We're going to be much more convinced that certain therapies work and others will not be used.
May I make a comment? I think the big problem in backache or low back pain and the leg pain, I very, very seldom we see a paper talking about the devices and also the rehabilitation. There is not a standard rehabilitation program. in any paper that I've read during the last 20 years about spinal cord stimulation. So I don't agree with the results, because in each country, each university, the methods for rehabilitation is not the same. This is a very important problem when we consider the result. And there is another problem. In Brazil, we talk about the regressão à média. I don't know how the term in English. When you put an electrode or you change it, the way by which the patient make his work, how he sits, how he have the foot, how is he coping with the rehabilitation, the drugs, and so on, changes very much. And this is not considered in the papers. So I think we should make new projects. I agree with all of you, but you should include also the other interference problems as the drugs, physical medicine, rehabilitation, and so on. Now a question about the magnetic stimulation. There is a question if someone has some experience with transspinal magnetic stimulation. We started a project 5 years ago using the method to select patients for spinal cord stimulation for pain, abnormal movements, and bladder disturbances. I believe that is a very efficient to select patients for spinal cord stimulation. This is our point of view. My paper was sent for publication last month, but it was the subject was spinal cord stimulation for abnormal movements. But I think it's very simple, cheap, safe, and there is a very good agreement between the result and the spinal cord stimulation using implanted devices. So it's a very good procedure by my point of view for selection of patients for implantation. What is your?
I would like to talk about this a little bit. I agree with Dr. Manuel and talk a little bit more, not about only the rehabilitation, but about the inflammatory process that some people has, and the way of life, and the people has some inflammatory basis. And when we are talking about modulate the inflammatory, it's systemic inflammatory basis. It's not only in the central nervous system, and it's like a systemic inflammation. So the studies has to include how are these patients and how inflammatory are then in these studies, and this is an important issue that you have to think about that.
Yeah, just keep in mind there's several studies. We had inflammatory arthritis included, including the sacroiliac joint problems and lumbar spondylosis and facet pain. Okay, give you an example. There's a certain percentage of those patients in census study, but also in several of new what we call virgin back studies that are ongoing right now. I participate in two, and so those data are available partially, but when it comes to the failure of the spinal cord stimulation, when the patient comes back to you and say, "Oh, my back pain is back," my fellows, without any, thinking, they don't even talk to me. They look at first if they have positive facet loading, SI joint problem, and they do injections and determine if partially that back pain is coming from other sources than discogenic or neuropathic. So that component of the pain is much more difficult to treat with spinal cord stimulation. We are talking in all of these discussions that we had about the spinal cord and peripheral nerve stimulation about other pain. We're talking about neuropathic pain, we're talking about discogenic pain. The inflammatory arthritis are, not easy to treat or partially treated with any form of the spinal cord stimulation right now. I couldn't see profound improvements and results that I was expecting to see.
I really found a very interesting comment about Dr. Vallejo talking about different waveforms and how they can modulate and which could be the best time for you to just turn off the stimulation since you started to make proteins change. So it makes me feel about the closed loop study because, this study came with tonic stimulation also, it's not high frequency, it's not burst, it's not different, but and they have a high level of evidence in this randomized study just close to DTM and high frequency. So these three waveforms, as Dr. Capral said. So I was thinking, suddenly everybody started talking about the kind of stimulation, the tonic stimulation, that they came with this closed loop and they get these good results also with this kind of stimulation. Maybe the way that they did was the way that they could modulate better this nervous system. So this is something that, give me some thoughts about
I would like to hear, so thank you, Tiago. So my thoughts probably are not as valuable as the thoughts of Leonardo because Leo was part of the original Avalon study. And I will love for him to share his experience regarding that study. But beyond that, I would say, yes, it is a very interesting study because it has shown positive results published in Lancet Neurology. And these results, I would say there is several things to think about it. One of the interesting aspects about that is if you think about how do you generate compound action potentials, is that you need to have, first of all, you need to have an active recharge balance, which is different to have a passive recharge balance. That means it is mandated for them that they have to have the cathode and the anode sent simultaneously. And I think that can have a very significant effect and I showed that in some of the slides at the molecular level, instead in terms of the expression of that. Now, the other thing is, can it be almost, I would say the study is very well designed. I don't have anything against that. The only thing that I don't like is that it's a double-blind study. And you will question why do I don't like double-blind study? And the reason I don't like double-blind studying is because there is only one person that was aware about everything. So the physician didn't know what was going on. The patient didn't know what was going on. But the clinical field engineer knew whether the patient was in one arm or another. And the clinical field engineer worked for one company. And the clinical field engineer had shares in one company. So, and that doesn't minimize the value of the study at all. I just want you to be aware that, first of all, in terms of the waveform, there is a difference between this active recharge balancing and the passive recharge balance that is commonly used in conventional stimulation. This is one aspect that I think could be important. And the other aspect is obviously how was the engagement of the programmer. And the programmer was the only one that knew what was going on at the end of the day. I don't think that affected the results at all. I know that Nagy was the chief medical officer, and there is few people in the world that I can trust more than Nagy, so I know everything was done correctly. And I think we are all eager to find out what will happen when that is brought to United States. I think they are still awaiting FDA approval. And I am very, very, and I think I would share that with Leo. What has happened since Leo was mentioning that he was leading the Bionex stimulation. He's leading everything in the last several years. I mean, any paper that is of value has been led by Leo. But that is fantastic. I mean, I'm all about science. I want people to move away, and I'm not talking, Tiago, at all about the experience that you have, which I think is very, very valuable. But there is certain companies that are promoting real-world experience instead of randomized controlled trials. And I think that is wrong, and I will tell you why. Because when you know the answer, if I look at the answers, and I know what I want to obtain, then I can formulate a hypothesis, and guess what? I obtain the results that I want. So I formulate what is happening right now with these all innovations and all these new randomized controlled trials that are going on, is that we will have a lot of evidence 3 years from now. When I say I mean we as physicians. We will have more tools to treat our patients. And I think that the companies should be encouraged by us to show real research to move the field forward, because otherwise we are not helping our patients. And I think, again, Leo is not one of the leaders, the main leader in the field. And I think we need research like the one that he is doing if we want 5 years from now to have answers for our patients. I don't know, Leo, do you want to comment on the Avalon?
Yeah. No, I totally agree with Ricardo. This was and you know that this was my advice with Stimgenics, but also with that the other arm of the randomized prospective trial should be left to other company if you're using their device. And the reason is you give the chance of that other therapy to be the best optimized as possible. And that happened with Stimgenics, that happened with the Senza 10,000 hertz study. But unfortunately, it didn't happen with other studies that we are quoting today. Well, with the DRG too. So if you let that happen, then you can really have much closer to the truth what happens in each arm of the study. Optimization will be proper, and they will compete each other like they did in the Senza study all the time. You know, we had the Boston arm of the study that we had reps coming in even off the schedule to reprogram those patients, just to show that the therapy is working so well. And so that type of the randomization I like the best, to be honest with you. You know, when you have in the hand of the same engineer both arms of the study, you know, all of us are humans, you know, and because we are humans and we are employed by the company that does the study, those bias can happen. The other thing that Tiago mentioned about Tiago, you mentioned this not feeling the low frequency stimulation pattern, and I can tell you from we did the most implants as far as per site in Evoke study with the Saluda, and the closed loop was actually pretty good when it comes to minimizing the effects of the paresthesias. Those patients really didn't pay attention to it, to be honest with you. Most of the patients, when you ask them, do you feel them? Yes, but it doesn't bother me. I think those surges, those baseline changes, you know, when the patients go down to lay down and sits up, that they don't have to adjust anymore their stimulator, or when they have a sudden surges, when they move suddenly, or when they keep the stimulation going overnight without any significant increase of the unknowing paresthesias and significant increase of the baseline, actually increase the compliance of the therapy.
Yeah. So I have a last question, very. If you that have the access to this therapy, because in Brazil we don't have all that you are having, but I know that in the US the insurance companies give you one shot. At least Golovac, when he came to our course, give a class, he said we have one shot to choose the type of waveform. So what do you think? What kind of shop you would give? Because you have 3 options that look like very similar. So what do you think? Do you think any of the 3 today? You know, I know that 5 years ahead we're gonna talk about other stuff. We will have more studies. We will have more, you know, experience. So maybe we change our minds. But what do you say today? Because this bothers me. You know, patients come to my office and I say, you know, what's the best therapy I have to offer to him? You know, what I would choose? Like I told in that first case, you know, patient have a system that is compatible with DTM. And I didn't have any experience with DTM to rescue a patient like that. And it works amazing, you know, and good in the help their patients. So what can you tell me about that? I know it's a difficult question, but, you know, just because it bothers me a lot, what I should...
We show some of the...
Good question, Tiago. In the last one, yes, great question.
We have the guys here, so we have to ask them. So, you know.
So we published just now with the University of Kansas rescue study with 10 kilohertz on more than 150 patients. It seems to be working extremely well, moving away from the traditional spinal cord stimulation. So definitely this is additional effect. Even those that were turned off for a couple of years, we were able to... I have now 9 patients that I moved from traditional stimulation to DTM. Every single one improved with more than 50% of the pain relief. So we are getting to this situation where choices have to be made maybe based on indications. Give you an example. We have a really great data on abdominal pain with 10 kilohertz. We have really good data on DTM with the back pain. We have fantastic data on 10 kilohertz with the neck and upper extremity, because that eliminates all of these issues with paresthesias. It still remains to be seen with the closed loop. I think some of these instances where you have a patients that are maintained with a certain type of severe foraminal stenosis and neuropathy and stuff like that, it may be that the closed loop will work better just because of the more tight adjustment to the therapeutic range. I don't have idea. I think when we get into the specific indications, we will have a more idea what we're going to pick. Right now, I'm picking 2 that are available to me and that I know that they have more than 80% success rate, and this is DTM and HF10. But, who knows? Tomorrow, we may be something else. The closed loop is not available in the United States.
So, Tiago, let me try also to answer your question. And first of all, let me tell you, as a scientist, I love uncertainty. And the final comment from Leo, I think, is so remarkable. We don't know what is going to happen in the future. And unfortunately, people come to these meetings expecting the right answer. And typically, they don't get it from me. So that's the bad part of it, because I like uncertainty. I don't like variables, though. I like what Jonas just wrote a few minutes ago about my comment in the sum of the parts that are only the parts, which, again, it refers that if you look that you're going to have an answer right there, it's going to be very hard. But your question has certain advantages because you are in Brazil. So one of the options that was proposed to you is not available to you, if I understand correctly. HF10, you don't have it. So that leaves the closed loop that also has shown good results. I think it will be, the original studies were not so good, but the last one was fantastic. So that you don't have it. So that takes 2 of those out. So I can scratch 2 of those options. So in my mind, you have the Abbott system, the Burst DR, and you already, and now let me put a little variable that you didn't include it, and it's the service that you get. And it's something that I think is very important. Who is the company that is serving you, and how much attention they will put to your patients? Because I think that is what you should, at the end of the day, you are the advocate for your patients. And you should pick up the one that will go the extra mile for your patients. Now, can they go the extra mile? Do they have the tools? The rep can be the nicest guy in the world, but do they have the tools? So in my mind, there is these three different variables. There is one company that can offer you burst, DR. There is another company that can offer you everything. They will come and say, hey, I can do everything. And you will say, yeah, but I need you to tell me, what do I do with my patient tomorrow? And their answer, because they have done zero research, should be like, I don't know, but I can do everything. So that one, I will scratch it right there, because they have not done anything to prove that they are worth for you to trust them. So that company is out. So now you have, again, the burst DR, or you have the DTM. The burst DR, you know, that has shown to be superior to conventional, but you also know that your chances are that you're going to have 39% success rate at 50% relief, or you will have 69% relief at 30%, depending what you want. But that's what you have, and 1.5 years later, if the tonic doesn't work, you're going to run out of battery and you're going to have to put a new one. While the other one gives you the opportunity to use conventional, it will also give you the offer. I don't like high dose, to be honest. I think that was a marketing response to HF10. So I think that it also offers you DTM. Now, I can tell you, though, that in the very near future, there is going to be more options for you, where you may be able to combine some of these different things in one single system. And we are working with that. But again, that will be my response. You will have, in my mind, you have two options, and it will be dependent on who is serving you, how good is the service, and at the end of the day, again, who cares what I say or anything? It's what your patient tells you. When I started my first presentation, Tiago, was why did I start this craziness for the last 16 years in my life? It's because I was frustrated and I didn't get the answer that you are asking me now to get it. And I obviously have failed so far to provide those answers. But I tell you, at the end of the day, it doesn't matter what anybody tells you in the podium. Somebody super cool comes and say, hey, you know, I'm awesome. I think this is the best thing. You are the one that is going to face your patient and tell them, you know, I'm really sorry it didn't help. And it's one shot. I only had one shot. I messed it up. I'm sorry. I went to this conference. I had beer, and it seemed like it was the best thing in the world, and it was.
Thank you. Very nice. Very nice. Very hard. I know.
As a scientist, Dr. Ricardo, as a scientist, I'm 95% confident that all of you were brilliant, were awesome, were outstanding, and this presentation is priceless. Thank you very much.
You just want to look for your daughter, my friend. That's what's going on. I saw her.
Thank you.
Thank you, guys.
Thank you.
Leo, what a pleasure to see you, my friend.
Thank you. Bye-bye.
Thank you all. Thank you all.
Thank you.
Bye-bye. Thank you very much.
Thank you, guys.
Bye.
As well. Thank you for having us.
Bye.
Por que ela tem que estar assim?